Welcome. Thank you for joining us. I'm Dr. Natalia Wagemans, Global Head of the Purina Institute. Today, I'm pleased to introduce you to the Microbiome Forum virtual event, Putting Microbiome Science into Clinical Practice, organized by the Purina Institute in collaboration with the top experts in microbiome, gastroenterology, veterinary medicine, and nutrition. Before we start our scientific program, I would like to introduce you to the Purina Institute. The Purina Institute is a global professional organization that serves as the voice of science for Purina, representing a diverse team of more than 500 scientists and pet care experts across a worldwide network of research and developmental facilities. The Purina Institute believes that science is better when it's shared. Through global collaboration, support the pet health initiatives and conferences and events like this webinar today, the Institute shares insights and discoveries about proven nutritional science. Nutrition influences every aspect of pet's life. As a part of the world's largest food and nutrition research organization, the Purina Institute aims to put nutrition at the forefront of pet health discussion. The Institute does not discuss products, only science. The Institute provides veterinary professionals like you with the science-based facts and information you need to have nutritional conversations with pet owners that can help dogs and cats live better, longer life. Now, you know a little bit about the Purina Institute, and if you would like to take more information, you can join us on the purinainstitute.com website and subscribe for our newsletter to get the latest nutritional updates. What is about the microbiome? Nestle and Purina are the leading organizations in the microbiome research. As a part of Nestle, Purina dedicated a long time to research in microbiome, prebiotics, and probiotics to evaluate potential probiotic strengths of value to veterinary medicine and pet health. Nestle has collecting bacterial strains since 1970s. The Nestle Culture Collection housed in Lausanne, Switzerland, contains thousands of bacterial strains that have been genetically sequenced and cataloged. Nestle published the first bacterium species genome sequence as well as the first lactobacillus species sequence from the human gut. As a pioneers in microbiome research to develop nutritional interventions that benefit pet health, Purina and Nestle made a certain discoveries that helps the veterinarians and pets to be better. For example, first direct comparison of the dog and human microbiomes to demonstrate the similarity between the microbiomes of two species as well as the similarity in their response in diet. Purina offered the first shelf-stable probiotic supplement, SF68, proven to promote a healthy immune system and provide dietary management of dogs or cats with diarrhea. Purina offered the first shelf-stable probiotic to be proven to help with anxious behaviors to dogs better cope with external stressors. And the event today is taking a time to look at the different aspects of the microbiome, science-based evidence, and how to put this into the clinical practice. As you could notice that we have two streams that you can join freely depending on the topic of the lecture. In some of lectures, you will see more academic aspects of the microbiome research, while in the other lectures, you will see more clinical and practical implications. At the end of the session, stay with us because you will have a unique possibility to join a questions and answers session with a panel of the experts who were building this program together and who will share with you science. You will be able to type your question online, and if you know whom you would like to address these questions, please indicate the name of a speaker. That will facilitate our conversations. And now I have an absolutely great pleasure to introduce a chairperson of this microbiome forum who put that program together with the eminent speakers and experts in area of microbiome, Professor Jan Suchodolski. Professor Jan Suchodolski graduated with a veterinary degree from the University in Veterinary Medicine in Vienna, Austria. After working for several years in a small animal specialty clinic, he returned to academia and received his PhD in veterinary microbiology from Texas A&M University for his work on molecular markers for the assessment of the intestinal microbiome. He is board certified in immunology by the American College of Veterinary Microbiologists. He currently serves as professor and associate director in the GI lab in Texas A&M University. He has published more than 340 peer-reviewed manuscripts in the field of veterinary gastroenterology and microbiome. And with this, I welcome Jan to give his keynote lecture on part of the whole, the role of the gut microbiome in the host health. Jan, the floor is yours.
Yeah, welcome everybody. It's my really great pleasure to be here. And so my talk will be really as a starting point and really to give an overview of all the different talks that's going to come. And it's about the part of the whole and why the microbiome really plays such an important role in host health, but also disease. First, my disclosure slides. I'm an employee of Texas A&M University, and we actually offering microbiome testing as a fee for service. But I'm also consulting or speaking for different companies. So why is the microbiome important? I think more and more over the last years, we discover that really gut bacteria play an important role. And some of them are really surprising. Like, for example, here's an example of a recent publication in a very high impact journal. And they've shown that some people that have so-called fatty liver disease. So it's a fatty liver disease. It's one of the most common disease phenotypes, liver diseases in human medicine. And it has been shown that some of those people have actually bacteria that can produce ethanol. So if they're on a high carbohydrate diet, they have some strains, they take those carbohydrates and ultimately convert them to ethanol. And that is believed to some degree contribute to this fatty liver disease. And even in the study, they showed even nicely that if you take feces or fecal samples from some of those patients, put them into a mouse model, and really you could induce liver disease in those mice. So again, showing that the gut microbiota is really plays an important role in some disease phenotypes that we until now didn't really well understand. On the other side, also like treatments that we usually always thought that they are to some degree beneficial or at least harmless. So getting wrong, antibiotics are super important for certain diseases. But there's more and more data coming out that antibiotic treatments, especially with broad spectrum antibiotic, can contribute to some chronic diseases that were now very prevalent. So for example, in this really huge study from Sweden, looking at several 120,000 patients, they showed that exposure to antibiotics is potentially associated with an increased risk for developing GI disease. So again, we have to overthink a little bit the benefits and the risks of antibiotic administration. There seems to be also some data out there that shows, especially in young childhood, in a certain young age, when the microbiome develops, when the immune system develops, antibiotic exposure also is a risk factor. It's a small risk factor. So again, I'm highlighting there's no right or wrong answer, but it just shows that when we use antibiotics, we most likely change the gut microbiota. And then again, it becomes a part of a disease phenotype. So again, we're learning more and more that the microbiome is clearly implicated in beneficial compounds, but also potentially, if changed, can be a contributor to disease. And why is that actually? Because really surprisingly, what we always thought, this is a waste product. So when you look at the GI tract and we look at a fecal sample, we always thought that's a waste product. But actually, it turns out that this is an important metabolic organ. It's immune, but also metabolically active. And the reason for that is if you cut through the lumen of the colon here, for example, that's in a mouse, but the same we see in humans and dogs and cats. What we see here in blue, that's epithelial cells, so that's the host. And here in red, all those cells here in red, those are bacteria. So macroscopically, what we see as a fecal sample is actually really full of bacteria on other microbes. and they are really alive and they're metabolically active. And so we need to understand that the gut microbiota is really part of our physiology. So everything, when you think about it, everything what's going to enter the gut. This could be nutrients and macronutrients, minor nutrients, but also drugs, medication. Everything what enters the guts is to some degree going to be changed, altered, or metabolized by bacteria. And again, along the entire GI tract, those metabolites are going to be ultimately absorbed by the host. And depending on the balance, so the balance of the microbes and the balance of what they're actually producing, we really have either pro-inflammatory or anti-inflammatory signaling. Ideally, it's obviously both. So again, it just shows us that this is important. But also what we're going to learn today a lot, and I think we really want to really emphasize that, we cannot separate the microbiome from our organisms. So very often what happens in the host, there could be inflammation, there could be other factors that's going to change the gut microbiota too. And again, so what we really need to understand is we cannot treat the microbiome by itself. There's always a reason gut microbes are changed. It could be dietary, but could also be some inflammatory components. And so ultimately, this is all one big organ. And for the future, multimodal approaches are really crucial. And that's why I think this connection, there's more and more data coming out that the guts and the gut microbiota really play a big role, obviously for health, but if there are alterations, there could be, again, different dietary substrates, but also some underlying disease process in the host that changes the gut microbiota. And ultimately, we have this really important axis between the gut microbiota, the circulation. We can have it, for example, in the kidney and so on. So again, we need to understand everything is going through the gut to some degree, and this becomes really a central role in many disease phenotypes, not just for the intestinal tract. So what I'm going to briefly talk today is really, what is the microbiome? Mostly focused on a few metabolites, because I think the more you read in literature, it seems very complicated. Yeah, every publication finds something. But I think it's like any other organ systems. If we reduce it to a few major metabolic pathways, and those are really the major ones, like the short-chain fatty acid pathway, amino acid metabolites like indoles and so on, and especially bile acids, I think that probably explains a lot of those effects of the microbiota on the host. I'm going to touch up also a little bit of dysbiosis, and actually really what is the most common reason, and that's really GI disease, underlying GI inflammation, or obviously antibiotic usage. So to understand that those are really the major factors, and trying to really highlight a little bit the relationship with the microbiome, dysbiosis, and systemic organ disease. So again, I think when you read literature, a lot of people say, well, it's so complicated, and it truly is. I'm sure it's like every other organ system. But I think when you really start looking at this in detail, there's a lot of pathways that really repeat itself. And again, I mentioned the short-chain fatty acid pathway is one important pathway. That's actually a pathway we knew for a long time. We have carbohydrates from the diet, mostly complex carbohydrates. And really, some key bacteria, and again, it's also important to probably start focusing on a few key bacterial species, because again, once you start reading literature, they're going to keep coming up. So it's really not as complicated as we may think, as long as we focus on one of those major pathways and major key bacterial species who do some of those functions. But important is that some bacteria really convert carbohydrates from the diets into so-called short-chain fatty acids. And again, also important to highlight, many of those metabolites really also depend on how much it is that. There needs to be a balance. In proper amounts, they're very highly beneficial. They're anti-inflammatory, and they also maintain intestinal barrier function. However, in abnormal concentration, if the ratios are different, then also they can actually activate some entropathogens. One important pathway is the tryptophan pathway from the diet. It converts by bacteria into so-called indole metabolites. And generally, again, it's anti-inflammatory, important for barrier function. But again, if it's in abnormal concentration, it can actually act as an uremic toxin. And the bile acid pathway, a very crucial one. And this is also one key species that we're discovering now in dogs and also in cats. It's really beneficial, it's Clostridium anonis, because it transforms those primary bile acids in the GI tract into so-called secondary bile acids. And again, in beneficial concentration, they're anti-inflammatory, inhibit pathogen growth, and so on. But again, if they're abnormal, they can either cause diarrhea or in too high concentrations that can also damage the mucosa. So again, everything is a balance. So a few examples, how do those metabolic pathways really work? So the short-chain fatty acid pathway is well established. I think we knew it for the longest time. And some of the short-chain fatty acids really, they bind to so-called G-coupled receptors. And those ultimately are anti-inflammatory. They can signal and can really improve barrier function, amongst others, so many different pathways. And again, Faecalibacterium, for example, is a very important beneficial species in dogs and in cats. There's also new features that we discover. Yes, so we know that those short-chain fatty acids bind to some of those receptors, and ultimately, if they activate many immune modulatory compounds. So generally, it is a beneficial pathway. But we're also discovering that that's why the microbiome becomes so central in systemic health, is that some of those receptors, for example, are now also discovered to be on other organ systems. So we always think about the gut, but as you can see, many of those receptors are also on the kidney and so on. Another important pathway, tryptophans. Again, as I mentioned before, tryptophan comes from the diet, gets converted by bacteria into different indoles, and generally it's a very important pathway. I think in GI disease, we're discovering more and more, if there's decrease in tryptophan, there might be some association that supplementing tryptophan, at least in mouse models, may be useful. However, again, balance is important. In certain conditions, bacteria can actually convert those indole pathways in so-called toxic metabolites like indoxyl sulfate and so on, with systemic negative effects on the host. So again, it depends always on where the host is. And again, the bile acid pathway is really one of them that is really surprisingly one of the probably key pathways. And the reason for that is that primary bile acids come from the liver, usually aid in fat digestion. Most of those bile acids get reabsorbed back in the liver for enterohepatic circulation, but a small percentage of bile acids gets actually converted by bacteria, so gut bacteria, into so-called secondary bile acids. And this is a beautiful example to show that really how microbiota is part of our physiology, because to our current knowledge, only bacteria can perform this conversion. So only bacteria, in this case, it's Clostridium nonis in dogs, also in cats, convert primary bile acids into secondary. And as you can see, there's a lot of effects. The proper ratio of those bile acids is crucial for signaling back to the liver. So for liver homeostasis, there's a lot of receptors like the TGI-5 receptor, for example, that is located in many different tissues. If it gets activated, and mostly those secondary bile acids are the strong activators, activation really downregulates pro-inflammatory signaling. We also know that's also important. Many of those metabolites really regulate the pathogens. So often we really see pathogens increased in GI disease, and we think that's the cause. But actually, importantly, because we have a dysbiosis, we have some of these metabolic pathways disrupted that ultimately leads to an overgrowth of pathogens. And finally, we also have, especially bile acids are very important also for insulin glucose secretion and so on. So again, as you can see, a pathway that is regulated by microbiota has many different systemic effects across the whole body. Here another example why bile acid receptors are important. As you can see, they activate different receptors and play a major role in glucose homeostasis, lipid metabolism, in motility, gallbladder filling, and so on and so on. Again, it's really a very important microbial pathway that has a lot of systemic health implications. And this is actually one we looked for several years now in the bile acid pathway. And this is work from Paula Garretta. She was back then a PhD student and a pathology resident. Now she is really successful pathologist in Brazil. And what she did, she actually in dogs, in healthy dogs, along the GI tract, stains for this important TGF-5 receptor. And you can see this is highly expressed in many different tissues, in goblet cells, colonocytes, macrophages, endoendocrine cells, ganglion cells, and so on. So again, clearly indicating that those receptors that are activated by bile acids are highly abundant in many different organ systems. So this is really a key pathway. And that really brings us also that, of course, it makes sense that we need to start looking at the microbiome. We need to understand, is it normal, abnormal? And obviously, when you look at the GI tract, it's a very complicated, long organ. So that's why it's also very difficult to truly appreciate the full microbiome along from the small to the large intestine. So there's really not one single test that allows the proper assessment of microbiome function. However, with time, we really, I think, we're able to, with many different studies and many different studies over time, we were really able to understand that some key pathways and also some key bacteria who do those pathways. So I mentioned, for example, Fecalibacterium, Clostridium hydrononis. So we developed assays for those. So we can really measure the abundances, and we actually can even describe reference intervals for those. And so we have developed a test called the dysbiosis index, who is really measures 7 bacterial groups. 5 of them are called beneficial, so they usually go down in disease. And some of them are more or less harmful, not necessarily directly harmful, but they are usually increased in dysbiosis and so on. And so we can really measure them. We can report their reference intervals across a huge population of healthy dogs around the world. We have a lot of different studies about that. But also more importantly, we can really combine the results into one single number. And so what you see here is the dysbiosis index. What you see here, each dot here is really the combined results of all those 7 abundances. And what it can tell us that we can really define to some degree where most healthy dogs lie, and they're below 0. Between 0 and 2 is probably like a small shift, a few healthy dogs later. And then above 2 is actually we're quite specific for that's an abnormal shift in the gut microbiota. But also we're discovering now that we need to kind of, interpretation is that we look at individual groups. If some of them are abnormal outside reference interval, that is a minor form of dysbiosis, and the index itself indicates how all those groups shift together. And we're also discovering more, and Dr. Torresen is going to tell you a little bit more about it. The higher the dysbiosis index, the higher the microbiome shift, usually it's a very negative predictor for treatment outcome. And because again, there is a lot of changes in the gut environment, and the microbiome really, dysbiosis reflects that in an increased dysbiosis index. So what are really major changes? So again, I mentioned antibiotic usage. The more we learn about studies, we need that antibiotics are really sometimes very useful, obviously, in many cases situations, but they definitely have an impact on the gut microbiota. So with this dysbiosis index, because it's a very highly reproducible test, we can really compare different studies, and we can really follow up patients over time. And you can see that different antibiotics like tylosin, metronidazole, broad spectrum ones, truly increase a huge shift in our gut microbiota. Most animals are going to come back over time within 1 or 2 months, but there's always a few animals that are going to stay abnormal. So in some animals with antibiotic usage, we can potentially induce a long-lasting change in the gut microbiota, especially in some of those important groups like Fecalibacterium or Clostridium hydrononis. Also in cats, as you can see, there's an assay we just finished validating. The paper is submitted. Similarly, below 0 is healthy cats. A lot of cats with chronic enteropathy have increase in dysbiosis index, again, decrease in Fecalibacterium and so on. And again, also in cats, antibiotic is a huge impact on the gut microbiota. So again, those are 2 major forms. Chronic GI disease and antibiotics have a huge impact on the gut microbiota. And why is actually GI disease so important? Well, think about it. This cartoon kind of illustrates a little bit how the microbiome is organized. So in a healthy gut, we should have a very well-organized microbiome, highly diverse, many different species. And they are separated from the epithelial cells by this mucus layer here in yellow. So this mucus layer separates the gut microbiota from the epithelial cells. Then we have a well-organized epithelium, and that's why also a normal functional immune system. Now, often when we look at GI disease and biopsies, we kind of always very much focus a lot on the infiltration. Is it what kind of cell infiltration is? But if you look really closely, we also need to understand that in GI disease, the inflammation is important, but also the architecture of the GI tract is very, very different. It means we have shortening of villi, we have loss of brush border, and so on. So again, it's, I think, important to understand that once a chronic GI disease has developed, it's not just the inflammatory component that's important. There's a lot of changes. And many of those changes we also discover now take a long time to normalize, if they normalize at all in some patients. So as you can see, in GI disease, we have always a dysbiosis, not always. In many cases, we can see a dysbiosis. In some patients, but not all, we have a decrease in this mucus layer. We have that because of that, we have attachment in some patients of bacteria to the epithelium. We have rupture of the epithelial tight junctions and so on, and an overshooting immune system. So again, in chronic GI disease, it's just that one part. There's a lot of different things that can go wrong. Many of them are very long lasting. So the function of the GI tract is abnormal. And that's what is really also drives dysbiosis. So it's not just one part. We need to always see this all together and ideally treat this all together. And so here's an example of fluorescence in situ hybridization in a healthy dog. Here you can see in red, that's the bacteria. And you can see in this black layer here, that's the mucus. This mucus is very, very important because it really separates out or the bacteria from the host epithelium. So that is a normal gut. In some dogs, but not in all dogs with chronic GI disease, it can look like this. Here in blue, you see epithelium. Then you don't see this mucus layer anymore. So we have a lot of bacteria that are attached to the epithelium, obviously inducing inflammatory components or pro-inflammatory signaling. So again, the problem is also a little bit that not every dog looks like them. So along the entire GI tract, a lot of different features, but understanding that many compounds in chronic enteropathy are wrong can help us ultimately thinking about what diets are needed. And again, important is that really, this is, I think, one that we probably, this functional aspect, once an abnormal GI tract is important to understand too, because once we have malabsorption, so this is again, the villus of a healthy dog. This is the work from Paula. And here in brown, you see the bile acid transporter in the ileum, and this one is responsible for absorbing of bile acids. And you can see how superficial these transporters are. And that's sort of the brushboard. And it's not just about bile acids. Here's absorption of, for example, glucose, fatty acids, and so on. So that's like really in a healthy dog. In a diseased dog, so many of this brush border is gone. That means we have also a lot of malabsorption to some degree. Many of those compounds ultimately going to be malabsorbed. They stay in the lumen of the GI tract, and that really also drives the dysbiosis pattern because bacteria like to feed on them. So again, understanding it's all a combination. There's not just dysbiosis. Dysbiosis in many cases really the reflection what happens in the gut environments. The microbiome is probably the first one who really reacts to changes on the brush border, inflammation, and so on, and often can be one of the first markers that there's something abnormal in the GI tract. And then ultimately, if this is abnormal, the gut bacteria can contribute to diseases because, for example, if there's glucose malabsorption in some patients, so this is in fecal samples, we see that some patients with chronic enteropathy have increased glucose, for example, bacteria obviously can metabolize it and can cause to some degree substances and again cause diarrhea. So again, there's a host part and microbiome part. We need to see them both together and ideally treat them together. And again, this is really probably my last slides for the moment for the first talk to understand, because I think we often think about the microbiome by itself. We think we can treat the microbiome by itself. Often we think about pathogens, we think that is the cause, but really what happens often, this interface between the host, the epithelium, and bacteria, that's where a lot of problems are starting. So often what happens in the host, inflammation can cause dysbiosis. Dysbiosis again can contribute to clinical signs of further GI disease. Then we have all of those components, changes in pathogens and so on. So understanding is that we need to think about when we see dysbiosis or enteropathogens, understanding that in most cases, the dysbiosis is a central part, but often, again, it's probably a consequence of those underlying disease processes here. And treating the underlying disease process is crucial. So it's difficult to treat just a microbiome. We need to treat the underlying condition, and usually not everything's going to normalize. So again, I think to summarize, and really many of those principles going to be discussed by some of our speakers later on, is that the microbiome is obviously very important. We need to see the microbiome as a metabolic organ, and that's why there's a lot of systemic health effects. So obviously it's important to keep the microbiome healthy. At the moment, we probably learned more than prevention is much better than therapy. But generally, we also need to understand we cannot just treat the microbiome by itself. We need to understand that dysbiosis is often the first mark of an underlying disease process that we need to identify and treat that, because if we just treat the dysbiosis, it's going to keep coming back because, again, it's the reflection of abnormal gut environments. But ultimately, we're going to discuss today from many speakers, what are pros and cons of diet, why diet is probably one of the most important reasons should always be considered dietary changes when we see an altered microbiome or dysbiosis. What are the benefits of prebiotics and probiotics? We're going to discuss, Dr. Unter is going to discuss the judicious use of antibiotics. Antibiotics are clearly needed in many diseases, but we need to understand in many, they're not important and might potentially have a risk factor. So weighing the benefits versus risks. And also we're going to discuss that actually fecal transplantation, we know a little bit more about in the last few years. There are a few case series, case reports, some retrospective studies, but there's also a lot of prospective studies ongoing. And I think we're going to see that fecal transplantation is going to be an option in some of those patients. Not in all, but it's definitely one option that we can consider in the future. So with this, I want to just acknowledge many of my collaborators that are really helpful in collecting all this data and understanding how the microbiome works. Without it, it would not be possible. Obviously, Garen supports, and I'm happy to answer any questions.
I have a pleasure to introduce our next speaker, Dr. Holly Lutz. Dr. Lutz is a project scientist in the Center for Microbiome Innovation and University of San Diego School of Medicine. And she is a scientific affiliate with Field Museum of Natural History in Chicago. She received her PhD in evolutionary biology from Cornell University, where she was affiliated with the Department of Ecology and Evolution. She studied the evolution of host symbiont system and immunology. Her research integrates natural history collection with metagenomic, metabolomic, and histological methods to investigate the evolutionary processes shaping these associations in animal systems. She also applies this method to clinical and experimental investigation into the role of bacterial symbionts in humans and disease phenotypes. Dr. Holly Lutz, please come and give your talk.
Hello, and thank you for the opportunity to speak with you about the microbiome and its role in intestinal inflammation and dysbiosis. My name is Holly Lutz, and I'm an assistant project scientist at the Center for Microbiome Innovation, which is part of the UC San Diego School of Medicine. Before I begin, I would like to mention a few things. First of all, I am not a veterinarian, nor am I a medical or veterinary clinician, although that might be my dream in another life. I'm an evolutionary biologist, and my research focuses largely on wild animals that you will rarely find as domesticated pets. I also want to point out that nothing I say here is intended to provide any clinical advice. What I'm hoping to achieve, rather, is the provision of broader context for understanding why and how some of the issues you face in the clinic occur. Great, so disclaimer out of the way, let's move on. In this presentation, I'm going to cover topics relating to the intestinal microbiome and enteric immunity to health in humans and animals. Many of you are already familiar with the concept of the microbiome. Just to define it again, the microbiome consists of any microbes, bacterial, fungal, archaeal, viral, or eukaryotic, that inhabit the host body. A common estimate is that the microbial cells outnumber our host cells 10 to 1, and our gut microbiome can actually weigh up to 2 kilograms, which is quite impressive. Kind of begs the question, are we human or are we microbe? We now also know from research that industrialization has led to a drastic reduction in microbial diversity in our guts. We also know that colonization of the GI tract begins at birth and stabilizes around the age of 3 to 5 years old. Of course, these facts are known primarily from observations of ourselves, humans. In this presentation, I'm going to cover topics relating to the intestinal microbiome and enteric immunity to health in humans and animals. Before I begin, I want us to zoom way out and remember that the vast majority of animal life on Earth is not mammalian, and that the animals on which we will be focusing today represent a mere fraction of diversity that has evolved. This is important because each unique animal lineage, from dogs and cats to ruminants, birds, fish, and so on, have evolved unique solutions to the problem of harnessing the power of microbial symbionts for metabolic health and immunological maintenance. That said, our goal here is to understand the links between the intestinal microbiome, immunity, and health in animals so that we can provide appropriate nutrition and health interventions for animals living under domestic conditions. To achieve this, we have to begin somewhere. And so today we will focus on the gains in knowledge that have been made by studying the mammalian GI system and associated microbes. In scientific research, we often rely on model organisms to identify general features of a system. In this case, In the case of the gut microbiome, humans are in fact the most well-studied mammals. We are essentially the model organism. So the majority of what we know about links between intestinal immunity and the microbiome is derived from human-oriented research. So it's important to keep in mind that what we think of about these relationships is likely to vary across different taxonomic groups. And so we shouldn't assume that clinical interventions for one group will necessarily translate for species of another, because of course, different animals are typically separated by millions of years of evolution, as you can see here. So with that, I would like to begin by reviewing some of the basic features of intestinal anatomy, immunity, and the role of the microbiome in healthy hosts. From there, I'll move on to discuss some of the conditions under which inflammation and dysbiosis can occur. And then I would like to finish with a brief look at some of the work that is being done to explore comparative features of the microbiome across disparate vertebrate lineages. In vertebrates, the gastrointestinal tract begins with the mouth and ends with the anus. Following ingestion and passage from the mouth and pharynx through the esophagus, food passes through the stomach, small intestines, and large intestine, each of which can be divided into further segments, as you can see in these diagrams. These anatomical features are largely conserved across vertebrates, but can vary dramatically in their morphology and measurements. Ruminants and other herbivores, for example, exhibit relatively long small intestines and a large cecum, which houses a large number of bacteria that assist in the enzymatic breakdown of cellulose and other complex carbohydrates. Carnivores, on the other hand, tend to have shorter small intestines, and insectivores, such as shrews, appear to be lacking a cecum entirely, as you can see in this fifth image on the right here. This anatomical variance is undoubtedly linked to diet and physiological adaptations to different lifestyles, and variation in the GI tract also has important implications for intestinal immunity. Looking more closely at intestinal anatomy, the stomach is a muscular organ where mechanical and chemical breakdown of food continues once passed on from the esophagus. The inner layer of the stomach, the mucosa, is densely packed with gastric glands, which contain cells that produce digestive enzymes, hydrochloric acid, and mucus. While digestive enzymes are secreted in the stomach, only a small amount of digestion actually occurs there, and no nutrients are absorbed. The digestive enzymes combine with the stomach contents and help to begin the process of breakdown of particles of food, with the enzyme pepsin beginning protein digestion. This combination of food, liquid, and enzymes passes from the stomach through the muscular pyloric sphincter and into the small intestine. The small intestine is comprised of three sections: the duodenum, the jejunum, and the ileum. The duodenum is typically the shortest section, and it is in this section of the small intestine that the pancreatic and bile ducts empty. The pancreas secretes digestive enzymes into the small intestine in order to aid in the breakdown of carbohydrates and other molecules. The small intestine is the main site for absorption and digestion of sugars, starches, minerals, fatty acids, and proteins, which have initially been processed in the stomach. It is also the location where absorption of the fat-soluble vitamins A, D, E, and K, calcium, and phosphorus occur. The small intestine transit time ranges from as little as 30 to 60 minutes up to as long as 8 hours. Anything that causes an increase or decrease in transit time through the small intestine will ultimately alter the absorption activities as well. To aid in nutrient absorption, the small intestine is covered by numerous folds of mucous membranes containing tiny projections called villi and microvilli, which further increase the total area for absorption. Absorbed nutrients are moved into circulation by blood capillaries or lymph channels. After passing through the ileum, the intestinal contents enter the large intestine and pass through the cecum, which contains enormous numbers of bacteria and acts as a fermentation site. The large intestine is wider and shorter than the small intestine and has a smoother inner wall. In the proximal or upper half of the large intestine, enzymes from the small intestine complete the digestive process, and beneficial bacteria generate short-chain fatty acids, or SCFAs, as a source of energy, as well as produce B vitamins, vitamin K, and some amino acids. The colon then absorbs the resulting nutrients in addition to water, which has accompanied the food throughout the digestive tract. Once the water is efficiently absorbed from the large intestine, any remaining waste which is not usable will be passed into the rectum and defecated. The mammalian gut is the primary site of interaction between the host immune system and the luminal contents, including not only food-derived antigens and toxins, but also the microbiota. Enteric immunity varies along the length of the GI tract, and microbial communities inhabiting the tract vary as well. Interactions and metabolic exchanges between hosts and microbes are mediated by enteric immunity, and a healthy balance facilitates intestinal homeostasis. The first barrier responsible for maintaining intestinal homeostasis is the mucosal barrier, which consists of both physical and chemical barriers. In the small intestine, chemical barriers include antimicrobial peptides produced by Paneth cells, which serve to segregate intestinal bacteria and intestinal epithelial cells. Peyer's patches bound along the length of the small intestine house lymphocytes that sample and respond to bacteria in the lumen, facilitating the production of regulatory T cells and anti-inflammatory responses. In healthy hosts, this communication between intestinal microbes and host immune cells promotes homeostasis and suppression of unnecessary inflammation. In the large intestine, mucus layers are thick and composed of two layers, the outer mucus layer and the inner mucus layer. The environment of the large intestine is largely anaerobic and houses a much greater number and diversity of microbes, which aid in the breakdown of complex carbohydrates. The intestinal epithelia are typically protected by the thicker mucosal layer, and fewer antimicrobial peptides are produced, allowing for the colonization and proliferation of a robust microbial flora. In the large intestine, secondary metabolites produced by the microbiota play an important role in host cell generation and differentiation, as well as mucus production and function. So to summarize this section, we know that physiochemical breakdown of food particles occurs primarily in the muscular and highly acidic stomach, where pepsin and other enzymes assist in digestion of large food particles. From there, food particles pass into the small intestine, where the majority of nutrient absorption occurs. Although some microbes are present in the small intestine, they are kept in check by the production of antimicrobial peptides in the dense array of lymphoid tissues like Peyer's patches and Paneth cells. Finally, any remaining undigested material passes into the large intestine, where a rich community of anaerobic bacteria aid in the digestion of complex carbohydrates while producing secondary metabolites that facilitate epithelial growth and differentiation. The environment of the large intestine is much more favorable for microbes compared to the small intestine, where fewer microbes are present, and the stomach, in which very few microbes can persist. And you can see here at the bottom of the slide that the pH ranges quite dramatically from the stomach, which is highly acidic, down to the large intestine, where it's largely neutral. So moving on to the next section, I'd like to cover a little bit about how inflammation and dysbiosis occur and what the mechanisms are underlying these. When the mucosal barrier is breached or the epithelial cell layer is otherwise damaged, the enteric immune system responds and inflammation occurs. In general, intestinal pathologies are the result of either acute or chronic inflammation. Although these are typically diagnosed as distinct categories, the lines are a bit hazy and symptoms are often indistinguishable. Acute inflammation occurs as a response to injury and is distinguished from chronic inflammation by the fact that the immune system is able to eliminate the cause of inflammation and repair any damage to the epithelium that has occurred. Chronic inflammation, on the other hand, occurs when the cause of epithelial or mucosal damage is not eliminated, leading the immune system to continually respond. The ability of the immune system to eliminate the cause of inflammation is the primary distinction between acute and inflammatory pathologies. And we now know that this ability is intimately tied to the milieu of intestinal microbes present in the gut. In the intestine, homeostasis is maintained via critical crosstalk between microbes and the host immune system. This occurs primarily in the small intestine. The landscape of epithelial villi is dotted with lymphoid mounds called Peyer's patches, where the majority of immunomodulation takes place. These are key sites for immune response to pathogens while promoting tolerance to beneficial microbes. Peyer's patches have a thinner mucosal layer than the surrounding villi and contain at their surface a number of different immune cells that serve different functions, including T and B lymphocytes, macrophages, and dendritic cells. Phagocytic M cells and dendritic cells sample microbial antigens such as bacterial lipopolysaccharides, short-chain fatty acids, and other secondary metabolites, and present these to T cells in the Peyer's patch. Under healthy conditions, a specialized subset of T cells, known as regulatory T cells, or Tregs, produce anti-inflammatory molecules, such as IL-10, or interleukin-10, which exerts a suppressive effect on immune cells within the lamina propria and upon the epithelial cells themselves. The function of Tregs is essential to maintaining intestinal homeostasis and immune quiescence. Defects in these T cells are a known cause of inflammatory bowel disease in both humans and animals. Mucosal damage, pathogen invasion, or even genetic predispositions can cause this process to break down, causing what is known as dysbiosis. And when this happens, it can result in gut pathologies ranging from acute gastroenteritis to chronic inflammatory bowel disease. When bacteria penetrate the mucosal layer, the Tregs, as well as the effector T cells, are triggered to begin producing pro-inflammatory molecules in an attempt to control the infection. These pro-inflammatory molecules can include interleukin-6, IL-12, IL-23, IL-17, interferon gamma, and tumor necrosis factor. Of course, these molecules and pathways may vary widely across different species, and we are still discovering new molecules and mechanisms that are linked to immunoinflammatory responses. In addition to this T cell activity, neutrophils are activated to capture and destroy the invading pathogens. And in this process, during this process, it can lead to additional tissue damage, and this propagates a cycle of inflammation and dysbiosis. This cycle of mucosal and epithelial damage triggering inflammatory immune responses can be caused by or can ultimately lead to major changes in the intestinal microbiota. Generally, what we see in mammals with inflammatory bowel disorders are a reduction in beneficial microbes, such as those in the phyla Bacteroides and Firmicutes, which has critical implications for the regeneration of new and healthy cells, as these microbes are responsible for the production of important short-chain fatty acids, such as butyrate, which is the primary energy source for cell growth in the intestine. Furthermore, a depletion of beneficial bacteria opens up space for harmful bacteria to proliferate. These include many species in the phylum Proteobacteria, which are opportunistic pathogens and which perpetuate the cycle of inflammation by continuing to degrade the mucosal layer. Lastly, when inflammation becomes chronic and dysbiosis sets in, host responses may begin including the sequestration of key nutrients such as zinc and iron that are required by beneficial and harmful bacteria alike, further hindering the return to a state of healthy homeostasis. Great. So we've covered the basics of intestinal anatomy, immunity, and the basics of enteric homeostasis, as well as some of the underlying mechanisms driving intestinal inflammation and dysbiosis. Now I would like to spend a few minutes exploring some of the differences and similarities we see in mammals across the evolutionary tree of life. As I've already mentioned, we are continually discovering new molecules and mechanisms that are responsible for promoting or suppressing inflammation in the intestine. And these discoveries are based largely on human research or the use of mouse models. Diverse species will likely have evolved diverse mechanisms for responding to these situations. And so it is important to remember that a solution or clinical intervention derived for one species might not be appropriate for others. As you can see here, and as I mentioned earlier, many taxa are separated by millions of years of evolution. And this means that selection has been acting on their immune systems for many years. And we need to take this into consideration. If we want to understand homeostasis in the animal intestine, we must first know what a healthy gut looks like. And to this end, a large amount of research has focused on describing the microbial symbionts that are present in the intestine. Due to the difficulty and complications associated with sampling the small intestine and the proximal large intestine, most research has focused on sampling of fecal material to understand which microbes are associated with different host organisms, as well as which microbes are associated with different disease phenotypes. Analysis of fecal microbiome composition has also played an important role in helping researchers and clinicians understand how to more accurately meet nutritional needs of pets and livestock. Here you can see the typical breakdown of the human fecal microbiome. It's about 20% actinobacteria, which contains some of the earliest microbes to colonize the human gut during infancy, which belong to the bifidobacterium. The largest group are the firmicutes, which include the bacilli and clostridia, followed by Bacteroidetes. And then the remaining microbes belong to many other groups. We can compare the human microbiome to the fecal microbiome of dogs. And while there are many similarities, we can see that a new group of microbes, the Fusobacterium, appears to be prevalent, while the actinobacteria, which are prevalent in humans, comprise a much smaller proportion of the canine microbiome. If we look at another carnivore group, the felines, we see that actinobacteria virtually disappear. And while Fusobacterium are also present, it is the firmicutes that really dominate the fecal microbiome among cats. In general, for humans, dogs, cats, and many other mammals, intestinal dysbiosis is correlated with a reduction in microbial diversity, particularly among the most common constituents of the gut microbiota. These typically include bacterial species belonging to the firmicutes and Bacteroidetes. In contrast, we often see an increase in unfavorable bacteria that are not usually present in high numbers, including species belonging to the Proteobacteria, which are facultative anaerobes, suggesting that in states of dysbiosis, there may be more oxygen available in the large intestine. The imbalance between beneficial and harmful bacteria has many important implications for host health. For example, as I mentioned previously, the reduction of butyrate-producing microbes, such as those belonging to the phylum Firmicutes, can negatively affect the regeneration and healthy differentiation of epithelial cells required for maintaining intestinal homeostasis. We are still in the infancy of understanding which microbes are responsible for which functions in the gut, especially when it comes to looking at different species. But advances in metagenomic and metabolomic methods are leading to rapid gains in this area of knowledge. Of course, what we think we know can often be disrupted when we look at mammals or other organisms from different branches of the tree of life. Take dolphins, for example. Earlier, I mentioned that the presence of proteobacteria in the intestine are typically a sign of dysbiosis and intestinal dysfunction. But in dolphins, proteobacteria are actually found to be a common constituent of the gut microbiome, making up to as much as 50% of the entire microbial population. Despite this surprising difference, dolphins are subject to many dysbiotic conditions that resemble those of terrestrial mammals, including irritable bowel syndrome, or IBS. In fact, I'm currently working closely with the US Navy's Marine Mammal Program to address some of the underlying causes of these symptoms in a subset of their highly trained and well cared for animals. Dolphins in this facility and others have displayed IBS symptoms that interfere with their performance and their quality of life. Anecdotally, Navy veterinarians have found that performing fecal microbiota transplantation has led to improvements in IBS symptoms. Given this information, we've teamed up with the Navy to explore whether FMT truly works, and if it does, how does it work? Despite the notable increase in proteobacteria that is normal for dolphins, it appears that some of the same culprits found in terrestrial IBS patients may be at work, including Fusobacterium and Enterococcus species, which we have found in our early analyses to be elevated in IBS dolphins. The study is still in progress, and we've now moved into phase 2. So keep your eyes open for updates if you're a dolphin doctor. Okay, I'd like to turn now to a completely different group of mammals, and one that is very near and dear to my heart, bats. Of course, bats have been receiving a lot of unwanted attention in recent years, and rightfully so. Bats are some of the most diverse mammals on the planet, second only to rodents. My research has found that they also harbor very unique microbiomes relative to other mammals we have studied. In addition to having a much higher abundance of proteobacteria than other mammals, even compared to dolphins, they appear to have a very low microbial load in the intestine in general. This suggests that perhaps bats do not rely on beneficial gut microbes the same way that many other mammals do. This is further evidenced by our discovery that bats do not appear to be co-evolving with their intestinal microbes, a term that we refer to as phylosymbiosis. This is very interesting and potentially really important because, as we've seen, the relationship between the intestinal microbiome and the host immune system is an intimate and highly regulated one. If bats do not need to maintain a specialized homeostasis with beneficial bacteria, could this mean that they have evolved different immunological pathways and adaptations for promoting or suppressing inflammation? Indeed, as we dig deeper into the genomes of the bats themselves, we've begun to learn that bats harbor many unique adaptations for suppressing inflammation. These include the losses of key gene families encoding functions for microbial recognition, as well as inflammatory cytokine production. It's hard not to wonder if these adaptations contribute to the diversity of zoonotic viruses harbored by many bat species. Lastly, what happens when we zoom way back out and look beyond mammals and at vertebrates more broadly? Another interesting finding in my research is that bats and birds actually harbor very similar microbial communities in the gut, wherein both are dominated by proteobacteria and both exhibit low microbial loads. This is why it is exciting to address these topics from an evolutionary perspective. For example, are these similarities the result of convergent evolution and adaptation to flight? That would make a lot of sense, but it could also be driven more largely by diet, as we've seen is the case in non-flighted mammals, such as herbivores and carnivores. Do bats and birds show similar immunological adaptations in the gut? And what does intestinal homeostasis even look like in a bat or a bird? These are questions I am now exploring in my research, and I hope that in the end, the discoveries we make provide helpful insights to the veterinary community and others responsible for maintaining animal health under controlled conditions. With that, I would like to conclude by summarizing some of the key take-home messages from this presentation. First, we now understand that for most, if not all animals, intestinal homeostasis is intricately linked to the symbiotic microbes present in the gut. This occurs because microbes provide services that are essential for host health, including but not limited to immune priming, nutrient acquisition, and defense against invasive pathogens. The determinants of a healthy microbiome are context dependent and will vary by species, diet, geographic location, and genetic background. In the same way, a healthy microbiome will be in large part determined by species' evolutionary history. And this should be taken into consideration when trying to understand the underlying causes and potential treatments for inflammatory bowel disorders. With that, I would like to thank you for your time and attention, and thank you to the organizers and all who are contributing to the field of intestinal health and clinical practice. With that, I'd be happy to take any questions. Thank you.
Our next speaker is Dr. Gail Charneski-Molden. She's a senior principal nutritionist at Nestle Purina and an adjunct professor of veterinary clinical medicine at the University of Illinois. She received her bachelor degree from Cornell University and master and PhD in animal nutrition from the University of Illinois. She served multiple scientific boards and multiple scientific societies during past years. Gail has published over 60 articles and abstracts in the area of pet nutrition. Her research focuses on probiotics, prebiotics, and the effect of nutrition on gastrointestinal health and microbiome. Thank you very much, Gail. We are looking forward to your lecture.
Hi, I'd like to thank the organizers for inviting me to speak at this forum. Today I'll be speaking about a subject that's near and dear to my heart. I've been a nutritionist at Nestle Purina for 30 years now, and for the last 20 years I've been working on probiotics and symbiotics and prebiotics. And what we found is there's a lot of misinformation out in the literature and in the world about probiotics particularly. So one of the things that we're going to do is to talk about how we might select a new probiotic strain, as well as symbiotics, and some of the misinformation that's out there about probiotics in general. So for the outline of my talk, we'll be going into factors that go into choosing an effective probiotic, the strain, the species of animal or that it was isolated from, stability of the probiotic, the proper dose. We'll talk a little bit about multi-strain products versus single strains, and then we'll end up talking a little bit about prebiotics and symbiotics. So the first thing we need to keep in mind is what is a probiotic? And the definition by the World Health Organization is that probiotics are live microorganisms which, when administered in adequate amounts, deliver health benefit for the host. And there are a couple key parts to this statement. Number one, they have to be alive. That's not to say that a dead microorganism might not work, but in order to be called a probiotic, it needs to be alive. Probiotics are not a generic type of thing. All probiotics are not the same as each other. The benefits are strain dependent. And by strain, I mean not species dependent. So if you have a Bifidobacterium longum, one strain of Bifidobacterium longum could have very different benefits than another strain. Stability is also dependent upon the strain. So some strains of bacteria are very stable, some are not as stable. And this is some work that was done by Ruth Noor and Rainer Simmering a number of years ago. And they were screening different strains of probiotics that were, these were particular ones were isolated from dogs. And in Ruth Noor's work, she had looked at 75 different strains of canine lactobacilli. Of those 75, only 16 actually had potential for probiotic activity. And in Rainer Simmering's work, he saw only 2 out of the 50 having potential. So by having potential for probiotic activity, what we mean is that they had potential benefits in vitro screening systems. And there was also screening to look at whether or not they might survive through the GI tract of the pet and whether or not they would be stable enough in the environment, the outside environment, in order to potentially be commercialized at some point. So some of the strains did not have potential benefits. Some of them were looked like they had benefits, but they were dying off in the in vitro GI system, and others were fine on those two, but they had to be refrigerated, or they might only have a shelf life of a week or something like that. So all of these things together need to be taken into consideration by the manufacturer when the probiotic strain is being selected. So some examples of types of information that might be available or should be available when you're selecting your probiotic strain. The first one that always should be available is a whole genome sequence. And this is very important for looking at safety of the probiotic. There are a number of things that are looked at depending on the species of bacteria, but we'll look for genes for antibiotic resistance and make sure that they're not atypical of the species of probiotic. You'll look for potential pathogenic factors, just anything that's that's unusual about that strain to make sure that it is safe to feed. You also look at potential to produce benefits. So again, this is dependent on what the benefit is that you're looking for. You might be looking for specific enzymes that are being produced by the bacteria. You might be looking for production of certain vitamins, neurotransmitters, amino acids, other bioactive proteins. So initially, this screening could be done in silico, just looking at the whole genome sequence and looking at what your targeted benefits are and whether that strain might produce those benefits. Another thing that can be looked at, again, typically this is done in vitro to start with, is antipathogenic activity. So some strains of bacteria will produce bacteriocins, and is this important to you? And are the proper bacteriocins being produced for that strain and for the end benefit you're looking at? You can also look at in vitro and in vivo effects on pathogen growth. And another one that we typically will always look at is the anti-inflammatory profile. So what is the cytokine production of this strain of bacteria? Is it an anti-inflammatory strain? Is it an immune stimulatory strain? So basically what you want to do is to evaluate the strains against the targeted benefit that you are going for. So the most important thing about choosing strains, or another really, really important thing about choosing strains, is to look at the targeted benefit of that strain. And I think that one important thing to keep in mind is that there is no single probiotic that can do everything. No probiotic is perfect. You can't have anti-inflammatory effects at the same time that you're having immune stimulating effects, for example. So when you're choosing a strain, you need to be very specific about what benefit do you really want this strain to confer to the pet. One example of this is, again, Enterococcus faecium SF68. The graph down at the lower left shows some information on canine distemper virus and antibody levels after vaccination. So this particular probiotic was fed to puppies from 8 weeks of age up to 1 year of age, and you can see there was a very nice antibody response to the vaccine in both the control and in the test puppies. But what started happening over time was that the antibody titers for the control puppies started to drop. So at 9 months and a year of age, you can see there was a significant difference in antibody levels between those two. And this is what we would expect from an immune stimulating probiotic. We expect some stimulation of the immune system, better antibody responses, more prolonged antibody responses to the vaccines. Because of this, it's not surprising then to see some effect on fecal quality during growth as well. And this is actually the graph over on the right is from a different study. And this study, again, was done with growing puppies. These particular puppies received the probiotic from 3 weeks of age. So as soon as they were starting to consume solid food up to a year of age. And you can see that what the graph represents is the fecal score of the puppies. The top line is the puppies who were on the probiotic, and the line below that, the yellow line, are the control puppies. On this particular scale, 100 means ideal feces all the time, and the lower it is on the scale, the softer the feces were and the closer to not being firm. So what you can see is that with the puppies who were on the probiotic, their overall fecal quality was higher, and there was also less, statistically less variation day to day. And again, this is something that we would expect from an immune stimulating probiotic. Excuse me. As I mentioned, you can also look at antipathogenic effects of probiotics. And a lot of times what we'll do is we'll do screening of the probiotic in an in vitro system, culturing it with the pathogen of interest. But you can also screen for potential effects on diarrhea. And again, this is something you need to be very specific about as far as what type of diarrhea are you interested in targeting. Are you interested in acute diarrhea that might be related to a pathogen? Are you interested in chronic inflammatory diarrhea? Because this could be a very different probiotic than one that's effective versus acute diarrheas. Are you interested in a probiotic that's effective against antibiotic-associated diarrhea? You can also look at other benefits, for example, behavior, disease, performance in livestock. Often we look at growth rate, we look at effects on fecal quality from day to day. And this is an example of Bifidobacterium longum, BL999. And this particular probiotic, the targeted benefit that we were most interested in was behavior and decreasing anxiety during stressful events in normal dogs, and also the ability to decrease anxiety overall in dogs who tend to be a little bit more anxious. And so again, we screened this probiotic in vitro. We looked at the whole genome sequence. We were looking for potential to affect neurotransmitters, to affect vagal response to stressors. And the results that you see in the graphs over on the lower right-hand corner show that there was, the lighter bar is the dogs who were on the B longum BL999, and you can see that there were significant decreases in cortisol levels during a stress, significant decreases in heart rate, as well as a significant increase in heart rate variability. And heart rate variability tends to be lower when people or pets are stressed. This is when you go into fight or flight mode and your body becomes very steady, getting ready for a threat. So an increase in heart rate variability is actually a sign of a better mental state and less anxiety. So there are a number of factors that can affect the efficacy of a probiotic strain. So we've been mentioning choosing a strain that has the benefits that you want, but there are also factors that will affect whether or not those benefits are expressed. And I think one thing that most people do not know about, unless you're very intimately involved in the production of probiotic, are the production aspects of this. So one question is, if you take a strain of a probiotic and produce it with different manufacturers under different conditions, will it have the same efficacy? And the simple answer to that is probably not. If you were to buy someone else's probiotic, take it, isolate the strains out of it and grow it up, it wouldn't necessarily mean that it's going to do the same thing. A good manufacturer is going to have a really tight quality system in place. This will ensure the absence of pathogens being introduced into the system, but also consistent genetics. A good manufacturer will have a master strain or a master lot of the strain, and then working seed lots that will come out of that. The master lot is kept separately, and what we'll do over time is you prove the identity of the production batches of the probiotic and compare those to the deposited strain. We need to make sure that the strain has not drifted at all as far as its whole genome sequence goes. And that will help to ensure that the strain is still beneficial and has the same stability that it had. The other thing is that production conditions will also affect the ability of the strain to have beneficial effects. And I've seen situations where you had a very beneficial strain that was having really nice physiological responses, and when the production system was changed, the fermentation media, or at what point the strain was harvested, or how long the fermentation was allowed to go on, you could actually have a total loss of all activity of the strain. So it's important to know how exactly to produce the strain, to make sure the system is well controlled, to make sure that the fermentation is always the same, and that verify the viability of the strain in those production conditions. There are a number of publications on this as well. Some of the things that can be affected by the processing or the production methods are the actual components of the cell. The structure of the cell can change. There's some publications showing that changes in the cell structure can actually affect the efficacy of the strain, and how you're fermenting it will actually can actually affect the actual structure of the strain, the cell. Gene expression can be affected by what's in the fermentation media, and also functionality, as we mentioned. So the pH of the media is important, how long it's kept at different pHs, the phase of growth when it's harvested, as well as the composition of the growth media. And I provided some example references for you here. There's quite a few additional publications on this as well. So the next question is, does it make a difference where the probiotic is actually harvested from? And we'll often hear people say, you know, if you're feeding a probiotic to a dog, the probiotic has to have originally come from a dog. Or if it's going to a human, it needs to have come from a human. The question I have is, where's the proof of that? When we look at colonization, so the argument that's typically made is that if you have a species of bacteria that's native to an animal, it's going to be able to colonize better and survive better in the GI tract. For most probiotics, we're actually looking for transient colonization, not permanent colonization. We want the probiotic to be able to go away when we stop feeding it, because it might not be necessary anymore, or it might be doing some things that you want it to do short term, but not necessarily want to have that probiotic live in the animal forever. So having transient colonization is fine. So that basically means when you're giving the probiotic, it's alive in the system, it will survive in the pet's gut, and it can do what it needs to do. But when you stop feeding it, within a couple of days, it'll probably go away. Probiotic strains can be perfectly efficacious with that type of colonization. So the other thing to keep in mind is whether or not your target is a target that would be present in all species. There might be certain benefits related to the physiology of the host that might only be relevant for one species of animal. But if you're looking at more general benefits, or benefits that would have a broader physiological activity across different species, then having a in-species strain or a strain that was isolated from a specific host is not as important. So again, some examples of this is, I'll use Enterococcus faecium SF68 because I'm most familiar with this probiotic. This is a lactic acid bacteria that has inhibitory effects against enteropathogens. For example, Salmonella, E. coli, Shigella. These are enteropathogens that can live in virtually almost most mammals could potentially have these. And this particular probiotic will kill these or have antipathogenic effects against these bacteria, regardless of what environment they're living in. This is also an immune stimulating probiotic, and again, will have this effect across multiple species of animals. It was originally isolated from the feces of a human infant, but there are numerous publications showing efficacy in humans, for example, before, during, and during antibiotic therapy in people as far as preventing traveler's diarrhea. There's a host of publications on livestock, different types of livestock, pigs, poultry, cattle, rabbits. I've seen information in zoo animals as well, having antipathogenic effects, growth promotive effects, effects on fecal quality. And there's quite a bit of data in pets as well, showing effects on fecal quality and microbiota stabilization. Personally, looking through the literature, I think this is fairly typical that when you have an effective probiotic, that'll be effective across a variety of species of animals. I'm not sure actually I've ever seen a probiotic that only works in one animal and only works in the animal it was isolated from. It's also important when you're selecting a strain that you want to maintain the viability of the probiotic. As we mentioned earlier, the definition of probiotic includes that they are live microorganisms. And there are a number of factors that can affect viability of probiotics. Oxygen, exposure to oxygen is very important, or lack of exposure to oxygen. The food matrix that the probiotic is present in makes a difference. The water activity or moisture present in the product, typically at a higher water activity, you will start seeing a loss of viability of the probiotics. Basically, when we have probiotics in a matrix, we want to have them kind of in a state of suspended animation. You don't want them actively fermenting unless you're in a situation, for example, if you're giving a yogurt to a pet or to a person, they would be alive at that point and active. And but those products only have a shelf life of a few days. The temperature is also very important. Typically, if you go too high, you're going to start seeing some loss of viability. The amount of time from when the probiotic is produced is very important, as well as the strain. So you can have different strains of the same species who have different viability. And when you start mixing these conditions together, you also will see a more rapid drop in viability. So for example, if you have a high temperature in the presence of high moisture, the probiotic will die off quicker. So it's very important to make sure that you're getting your probiotic from a manufacturer who is looking at the viability of the probiotic to make sure that the probiotic is still alive at the time it's fed to the animal. Okay, it's also another thing that I've heard a lot is, in order to be efficacious, your probiotic you have to be feeding 1 billion or 10 to the 9th bacteria per day. So is this true? Do you really have to have 10 to the 9th bacteria per day? And if you have a probiotic on the market that has fewer bacteria, does that automatically mean it's not going to be as effective? There have been very, very few dose response studies published on any probiotics. Most of the probiotics on the market that are efficacious, the efficacy studies were done with 1 billion bacteria per day. And if there was dose response work, it was not published on it. So basically the manufacturer would pick a dose that they thought would work. And I could tell you, we've done this as well. You feed that. If it works, then you might just go ahead and launch the product with that level of bacteria and not go back and look at whether or not a lower dose would actually work. So again, here's an example with Enterococcus faecium SF68. This is a kitten trial, and the probiotic was fed to these kittens from weaning to 1 year of age. And we had either control product, 5 times 10 to the 7th CFUs bacteria per day, 1 times 10 to the 8th, or 5 times 10 to the 8th. In this particular study, there was an outbreak of diarrhea, and the incidence of diarrhea was lower in the kittens who were fed SF68 than in the control. And these kittens also recovered much quicker than the ones who did get sick recovered much quicker. What we saw when we looked at the doses of the probiotic is that the 5 times 10 to the 7th was just as effective as the 5 times 10 to the 8th. And the product we have in the market has 5 times 10 to the 8th. This gives us a little bit of leeway in case there is some loss of viability. But again, you can see this is quite a bit lower than the 10 to the 9th bacteria that often you'll hear have to be present in a probiotic. So, basically the important thing is to make sure that whatever dose is in the probiotic, that's the dose that the efficacy studies were done at. Another thing that you'll often hear is that multi-strain probiotics are better than single strains. Having a variety of bacteria in the probiotic might potentially enhance efficacy. So the question is this true? Possibly. I'll say yes, no, maybe. It depends on what the bacteria are. You need to look at the targeted benefit, and the strains that are in the product need to be picked based off of what they do in the system with the other probiotics, but also how they might affect that benefit. So if you just randomly say, okay, I'm going to take 3 bifidobacteria, lactobacilli, and an Enterococcus, put them together, that's going to be better than any of those probiotics alone. This is not necessarily true. So if you're looking at, for example, an immune benefit, if you take an anti-inflammatory strain and mix it with an immune stimulatory strain, what effect on the immune system are you going to get? You can't have immune stimulatory effects at the same time as anti-inflammatory. One of those will predominate. So which benefit are you looking for, and what happens when you mix these strains together? What does the data say about this? Are there actually studies looking at efficacy of multi-strain products versus those individual strains to make sure that they actually work better in combination than alone. And there's very, very few publications that are actually comparing the probiotic to its individual component strains. There are some publications that show efficacy of multi-strain probiotics versus a single strain, but the single strain is not a strain that was in the multi-strain product. So it could be that the multi-strain might be better than that single strain for that particular use, but it doesn't mean that it's necessarily better because it has more than one bacteria in it. So as I said, there's few that are comparing multi versus single strain. There's even fewer publications that compare a multi-strain product to its component strains. Here's a couple examples of these. In the first publication, basically none of the combinations, they looked at a couple of probiotic bacteria and looked at combining the bacteria and found that none of the combinations had additive or synergistic effects on the cytokine profiles that they were targeting. And actually the cytokine response of the mixed product was actually closer to an average of the responses of the individual strains, which is really what we would expect to happen in most cases. And so by having the combination of bacteria, they were actually blunting the effect on cytokines. So there was a really nice review article that was just published, and the conclusion really was that multi-strain probiotics might be more effective than individual component strains if the effects are synergistic, but there's really a lack of research in this area. And I know when we've looked at multi-strain products, we have some under development right now. We've selected for strains that have very specific benefits and have in vitro data showing, or in vitro data showing that the combination of the strains actually is better than the single strain, than each of those strains alone. So it really just depends on what the product is. You can't make a blanket statement that multi-strains are always better than single strains. So when we look at the product label for probiotics, there are a couple things that we can get a hint from as far as what's on the label. First of all, the ingredient statement should include the specific probiotic or probiotics that are present. There should be a guarantee of live bacteria, and that guarantee should be at the end of shelf life. Occasionally, you'll see a product that says guaranteed at the time, it has this number of bacteria at the time of manufacture. That's always a red flag to me, because we're not feeding the probiotic in the factory where it's being made. We're feeding it maybe months after that. So we need to make sure it's alive at the end of shelf life. If it's a blend of probiotics, the guarantee should be for each genus. So I'd like to finish up just talking a little bit about prebiotics and symbiotics. So as we mentioned, probiotics are beneficial bacteria that are fed to the animal, whereas prebiotics are food for the beneficial bacteria that are already in the gut. So in order to be considered a prebiotic, it needs to be a non-digestible food ingredient that is beneficially affects the host. And typically these are broken down by the bacteria that are present in the gut. They're resistant to digestion in the upper GI tract so that they can actually be food for the bacteria lower down in the GI tract. And they provide a physiological benefit. I think the thing to keep in mind here, because there's some confusion about what is a fiber versus what is a prebiotic. All prebiotics are fibers, but not all fibers are prebiotics. A symbiotic, on the other hand, is a mixture of more organisms together with a fiber or a prebiotic or another compound that has an effect on the host. There can be a complementary product, so you have different ingredients that have different benefits, but they don't interact codependently, or you can have a true synergistic effect where a substrate that's in there or the prebiotic is actually used by the co-administered probiotic. And either of these situations can be true. So just in conclusion, when we're looking at development of probiotics and symbiotics, we need to have a very defined target. The strain of bacteria and or fibers should be chosen based on that specific target. The production of the bacteria should be optimized to optimize efficacy as well as stability, and how these bacteria are produced does make a difference on both of these. There should be efficacy studies done with that specific strain of the probiotic, and the dose should be based off of efficacy studies, and higher is not necessarily better. You just need to make sure that it works at the dose that it's being fed at, and that that dose is still present in the final product. So thank you for your attention, and we'll be having a question session at the end of the set of presentations.
Our next speaker is Dr. Jörg Steiner. Dr. Steiner received his veterinary and medical degree in Munich, Germany. He achieved board certification with the American College of Veterinary Internal Medicine and the European College of Veterinary Internal Medicine. He received his PhD from Texas A&M University on his work on canine digestive lipase and their use for the diagnosis of gastrointestinal disorders in dogs. In 2016, Dr. Steiner was named as the Dr. Mark Morris Chair in Small Animal Gastroenterology and Nutrition, and is involved in a wide variety of research in small animal and comparative gastroenterology. He has authored and co-authored more than 350 peer-reviewed articles, 100 books chapters, and 470 research abstracts. And now I give the stage to Dr. Steiner. Let's welcome him.
Welcome, everybody. Jan and I will be talking about diagnostic evaluation of the gut microbiome. But before I get started, I just need to briefly share my conflict of interest disclosure. I am the director of the gastrointestinal laboratory at Texas A&M University. And of course, we offer diagnostic testing on a fee-for-service basis. I'm also a paid consultant for a variety of companies: IDEX Laboratories, Nutramax Laboratories, ISK, Glykos Bio, and Bond Pet Care. And I'm a paid speaker for a variety of companies and get some grant support. None of what I have to say today should be influenced by any of this. The gastrointestinal microbiota is really a collection of all the gastrointestinal microbial organisms. And most people, when they talk about the microbiota, they think initially just of bacteria, but that's really not just it. There is also viral organisms, there is fungal organisms, there is archaeobacteria, so there's a wide variety of different microorganisms, but of course the ones we do think mostly about are bacteria. It also, and that sometimes even slips my tongue, it was known to be as the gastrointestinal microflora, but of course, we know there's no plants growing in the GI tract, and so we don't try to use that term anymore, so I apologize if I use that term on occasion, but it is really the microbiota. Now, the bacterial ecosystem, then when you focus more on the bacteria, are all the bacterial cells in the GI, mammalian GI tract, and they outnumber the actual animal cells in the GI tract, which is quite phenomenal if you think about it. And in fact, they account for 95% of all cells that are in the GI tract. And they represent as many as 500 to 5,000 different species. Now, that's kind of interesting because if somebody would have talked about this 10, 20 years ago, they would have maybe said, well, there's maybe usually 10 or 20 species in the GI tract, because at that point, all we knew is what we could culture from the GI tract. And of course, that was very, very limited. Now we know so much more, and we're much better able to understand what all these different bacteria are. Now, why should you and I care? We're clinicians, we care about dogs and cats being healthy and having no diarrhea. It is because the intestinal microbiome changes with disease, and we'll get back to that in just a second, whether it is this way or the other way around. And an altered microbiome, or you could also call it dysbiosis, has consequences. It expands the abundance of pathogenic and potentially pathogenic bacterial species, and there are some functional changes. So, for example, the most important one we are aware of these days, but there's many others, is a failure of conversion of primary to secondary bile acids. And these functional consequences then in turn can either perpetuate or worsen the disease process. Now, intestinal dysbiosis and disease of either the GI tract or the whole host have a kind of a chicken and egg relationship, which is the GI host and GI and host disease have an impact on the intestinal microbiome, but the intestinal microbiome may also have an impact on GI and host disease. And so it's really not entirely clear which one, is the more important one, which one comes first, and which one comes second. And maybe it really doesn't matter in the end. We just, as long as we understand that both of them are very important and they are interrelated with each other. Now, briefly, you can sort of differentiate different types of bacteria. And this is not an ideal, differentiation But just sort of philosophically, you can say, well, there's pathogenic bacteria, there are the beneficial bacteria, and there are the ones in between, which we call potentially pathogenic bacteria. Let me expand on that real quickly. So the pathogenic bacteria are bacteria that anytime they're there, they cause disease. And, you know, we think about salmonella like this, and in people, certainly salmonella, you would consider that a true pathogen. But, you know, in dogs and cats, that becomes a little bit more difficult where, you know, they may or may not cause disease every time they're there. Certain Campylobacter species belong to there, and certain E. coli species belong to there. So these are bacteria that really always cause disease. And on the very opposite side of the spectrum, you have beneficial bacteria. These are bacteria, good bacteria, as we also would call them. These are bacteria that do not cause disease. In fact, they're important. Their presence is important for the production of nutrients, for inhibition of other of pathogenic bacteria. They ferment fiber and prebiotics to nutrients that the gut then can use. They inhibit the pathogenic bacteria. And then you have the potentially pathogenic bacteria. These are bacteria that usually don't cause disease, but under certain circumstances, they in fact can cause disease. And again, certain Campylobacter strains belong to that. Certain Clostridium species belong to that. So you really have these three classes of bacteria. And so that concept is, you know, that bacteria on one end of the spectrum, the good bacteria, we want them there. And then the other spectrum they're the bad bacteria, they're the pathogenic bacteria, and we really don't want them there. And it's sort of the balance between of all of us, whether you have a host disease or host health. Now, the importance of the intestinal bacterial ecosystem is also quite clear. And that there's a lot of studies in germ-free rodents that show that you have a thinner and less well-hydrated intestinal wall if you're lacking the normal bacteria. You have a less well-developed immune system of the mucosa if you don't have the healthy bacteria. You have, in fact, a decreased mucosal surface area and an altered gastrointestinal motility if you don't have a normal bacterial microbiome. And so the beneficial bacteria are really crucial in a normal GI tract development, in a normal GI tract immune system development, and also in overall host health. Now, one important thing, of course, is we want to be able to evaluate intestinal microbiome. And here's a list of all the things that have been proposed, and we'll come back to that list at my end of my segment here, is to say, well, what of these things is actually practical and usable? But here's a whole list of things that have been proposed to be used to evaluate the intestinal microbiome. You have the quantitative culture of chyme or duodenal juice, which is really considered the gold standard, and we'll get back to that in just a moment. And then we have a wide variety of indirect tools, such as serum folate and serum cobalamin concentration, fecal culture, serum concentrations of unconjugated bile acids, hydrogen breath testing, intestinal permeability and mucosal function testing, short-chain fatty acids in duodenal juice or in serum, urine indican concentration, C13 chyloglycine testing, or sequencing and dysbiosis index. And hopefully, when we're done with this, you understand why there is only very few of these that actually are clinically useful. So let's first start with the quantitative culture of duodenal juice. And the first problem when you do that, when you try and do that, is the collection of such juice. And there's two ways you can do that. You can go to surgery and take a needleless syringe and try to aspirate fluid from the intestinal loops. That, of course, is very invasive and not always successful because you have to find a pocket of fluid. Or you can go ahead and you do endoscopy and you aspirate through fluid through the biopsy channel. Now, anyone who's ever tried to do that will understand just how frustrating that is because it is very difficult to aspirate any kind of fluid through the endoscopy channel, mostly because, you know, the animal's under anesthesia. You have usually the production of intestinal fluid is decreased during such anesthesia. And so it's very frustrating to collect a sufficient sample. And then you have to immediately go ahead and plate this sample in multiple dilutions, in multiple setups of different growth agars, growth media, and you have to grow them both at aerobic and anaerobic conditions. And then you have to take all of these cultures that you grow and speciate them. And you can imagine what a tremendous amount of work this is. And I can tell you, I was trying to do a research study about 10, 15 years ago at the Animal Medical Center in New York. And we were looking for labs first in New York City, both veterinary and human laboratories that would be willing to do this for us. And we're not even talking about cost here, but just willing to do this. We couldn't find one in New York City. We couldn't find one in the state of New York. In fact, we couldn't find one in New England. And so we finally aborted the mission because we simply were unable to find a lab willing to do this, because it's a tremendous amount of work. And then, of course, also, it really depends on how fast you were able to plate it. It depends on, the actual growth media that are being used. It depends on, the growth conditions. So there's a lot of variables that have a huge impact on what you actually grow in the end. So even though considered the gold standard, you can understand it's really not a very good gold standard, because it really doesn't give you very good and reproducible answers. And it's expensive and unpractical under practice conditions. So certainly not something you want to do in everyday practice. You could measure serum cobalamin concentration, and it's been shown that serum cobalamin can be decreased in small intestinal dysbiosis. Now, if you remember, we don't want to go through the entire physiology of how cobalamin is being absorbed, but it's important to remember that cobalamin is being absorbed in the ileum for the most part. And so the idea being that if you have too many bacteria, let's just call it too many for a second, dysbiosis is so much more than just too many, but let's just say you have too many bacteria that utilize cobalamin, then there's less cobalamin available for the absorption into distal small intestine, i.e. the ileum. And so you can imagine that if this dysbiosis happens to be there for a while, you ultimately will deplete your stores of cobalamin, and your serum cobalamin concentration will go down. And that has been described, but it also has been described that the sensitivity of doing this, of measuring a decreased serum cobalamin concentration for small intestinal dysbiosis, is only approximately 50%, which of course is not very good. You can also measure serum folate concentration, and serum folate can be increased in small intestinal dysbiosis. And again, we can't go through the entire absorption of folate here, but folate is absorbed in a very proximal portion of the GI tract. And the idea is that many bacteria actually synthesize folate, and we all know this because we use antibiotics that inhibit folate synthesis and thus kill the bacteria. So we know that folate, is being produced by the bacteria, and many bacteria in the GI tract do this. And so if you have too many bacteria in the small intestine, again, that concept of too many bacteria is not a very good one for dysbiosis. We'll get back to that later on. But certainly when that happens for a long period of time, you absorb more folic acid, and thus your serum folate concentration will ultimately go up. Now, fecal culture has been used by some people to try to have an estimate of the bacteria that are present in the small intestine. And the first thing I would tell you is that the bacteria that are in the small intestine are, of course, not the same that are in the feces. I'll show you that based on a study, but also that it has a horrible reproducibility. There's a study that was done in Munich recently where a fecal sample was sent to 3 different veterinary laboratories for evaluation for culture. And the results really showed that there was a wide variety of what they reported of the presence of bacteria. So it is not reproducible, it is not reliable, and it probably does not reflect at all what's going on in the small intestine. And I'll show you this is a little bit confusing here, but what I want you to first show you, this is basically simply looking at the genetic data of the bacteria that are present. And so each one of these bands here, if you look at this one right here, this individual, each one of these bands represents a different bacterial species. And so, of course, the bigger the band, also the more of it, the more abundance of this particular species do you have. Now, what this dendrogram also done, it shows you how closely related these are. And you can imagine like this lane here and this lane here, they almost look the same. And so that's why they're right next to each other, because they're very, very similar to each other. Then the other thing you need to notice are the colors here. The red being duodenum and jejunum, yellow being ileum, and blue being colon and rectum. And so here, this is basically information from healthy dogs, taken from healthy dogs. And it basically is a full spectrum, meaning this, where it ends here, it continues right here. So you have to imagine you're sitting in the middle and you're looking around in a 360-degree fashion. And what you very quickly see is that all the blues sort of are gathered together, and all the reds are sort of gathered together. And in between, you have the yellows. And what that means is that the microbiota between the colon and rectum on one dog are much more similar than what's in the duodenum and the jejunum on the same dog. Because, you know, the one dog, the duodenum and jejunum would be here, yet its colon and rectum would be here. And so you can see that there's no relation to what's in the colon and rectum as far as speciation goes to what is in the small intestine. And we'll get back to that later on, where when we look then on certain types of bacteria and basically forget about the rest, then that becomes different. But if you just look at the global picture, then that's unfortunately the case. Now, in the late 80s, early 90s, people started measuring serum unconjugated cholic acid, or SUCA. And certainly in humans, it was shown that small intestinal bacterial overgrowth, which is very defined by the number of bacteria that are present in total and the number of anaerobic bacteria that are present, have an increased serum total unconjugated bile acid, as well as serum unconjugated cholic acid. And in an initial study, then Melgreca et al. Showed that mildly to severely increased SUCA concentrations in 9 of 10 dogs with what they at the time called culture-proven small intestinal bacterial overgrowth. But as they studied this further, this did not appear to be very reliable or reproducible in a larger number of dogs, and so this concept was abandoned. Hydrogen breath testing was described by Washabau in the mid-80s, and basically he measured hydrogen in the expiratory air, suggesting an intestinal dysbiosis. And here you have basically what you do: you give a carbohydrate to the animal, you let it be, a non-digestible and absorbable carbohydrate, and then it gets fermented by the bacterial microbiota. In a healthy animal, what you can see is that initially no hydrogen is being produced, but then once that carbohydrate reaches the colon and it starts to be metabolized, fermented by these bacteria, you have a production of a small amount of hydrogen. So you have a late, not very high, but you have a peak in the hydrogen breath testing. And in theory, then when you have small intestinal bacterial overgrowth, meaning too many bacteria in the small intestine, what happens is the carbohydrate gets fermented early on in the small intestine, and you have this early peak in hydrogen in the breath. And you can see this here, many bacteria here in the small intestine, they produce the hydrogen, and you have this early peak. Now, the problem with this is multifold, but one of the biggest issues is, how do you collect breath in dogs? Well, you know, in people, it's very easy. You let them breathe into a little bag. Well, in dogs, when you put a mask on them like this, many dogs just stop breathing altogether, and CO2 accumulates, so their hydrogen also accumulates, and you can't measure anything. Or they start hyperventilating, where they actually move air back and forth. And in fact, if you measure the CO2 in there, its CO2 concentration goes way down, because they don't no longer give you expiratory air. And so it's a huge problem. And then, of course, the dead space in the mask. And we sort of then designed a little mask adjustment that would help us collect the hydrogen and, in a collection system. But the truth was, after measuring several hundred dogs, their expiratory air, we had to admit that it was simply not reproducible, and we stopped this altogether. We were also measuring intestinal permeability at some point. That had been shown by Roger Batt and his group, that it is abnormal in dogs with small intestinal bacterial overgrowth. And also the function, meaning the absorption of some sugar, is abnormal, but again, very difficult to do, very cumbersome. You have to give a sugar solution by gavage to the dog, and then you have to collect all the urine. So they basically have to be in a metabolic cage, and then you have to measure these sugars, which in itself is difficult and not highly reproducible. So this really did not turn out to be a good marker. Short chain fatty acids in either duodenal juice or serum. Now, the nice thing about short chain fatty acids is they're the main metabolite, the main nutrient that comes from fermentation of carbohydrates by these bacteria in this gastrointestinal tract. And theoretically, you can measure them, you can practically measure them, but these assays have not been validated for use in dogs. And there has not been much research done with that. So currently, certainly, this would not be a clinically useful diagnostic test for small intestinal dysbiosis or intestinal dysbiosis. Urine indican is another metabolite, and basically it is based on intestinal bacteria metabolizing either tyrosine or tryptophan from the diet and producing 4-hydroxyphenyl acetic acid, phenol or indican. And then theoretically, you can measure that in the urine, but this has not been yet evaluated in dogs. And again, not many labs are measuring this, so this is probably not something that is a reasonable diagnostic test. And finally, here we have 13C cholyl glycine testing, and the principle of that is fairly simple. You basically give the dog by oral administration C13 cholyl glycine, and so what this is, it's basically a conjugated bile acid. So it is a primary conjugated bile acid, and basically it is marked with C13, which is a non-radioactive rare carbon isotope. And what happens then, it gets down into the GI tract, and in the small intestine, it gets deconjugated, and the C13 glycine then is being absorbed. That then gets oxidized and ultimately metabolized to C13 CO2. And so the more deconjugation you have, the more C13 glycine you're going to get. I mean, glycine being the amino acid that cholyl glycine was, or the cholyl cholic acid was being attached to. And then the CO2, of course, gets moved into the blood and gets transported in different ways. But the relationship of C13 CO2 and C12 CO2 stays the same. And so basically, the more deconjugation you had, and the more C13 CO2 you had in the small intestine, the more you have ultimately getting C13 CO2 in the expiratory air. Now, we just talked about that you can't measure breath very well in dogs, but what you can measure is actually the blood. And basically what you do is you take a blood sample, you add some very strong acid to it, and that drives the bicarbonate out of the blood sample. And now you have basically a breath sample above the blood sample, in which you then can measure the ratio of C13 to C12. Again, remembering that the more deconjugation you have, because the more bacteria you have that can deconjugate bile acids, the more C13 CO2 you have in relation to C12 CO2 in this expiratory air that is above this blood sample. So when we go back and summarize what we talked about, how can we evaluate the intestinal microbiome? Well, we said quantitative culture of chyme and duodenal juice, not very reliable, very cumbersome to do, very hard to do, certainly not, while considered a gold standard, certainly not a practical, certainly not a clinically useful diagnostic tool we should be using. Serum folate, then the indirect diagnostic serum folate and cobalamin concentration. We talked about that, yes, they do work, but the sensitivity of these diagnostic tests is not very high. It's estimated to be approximately 50% for both increased serum folate concentration and or a decreased serum cobalamin concentration. Now, the fortunate thing for serum folate concentration is that there's really nothing else other than supplementation by the owner. Several owners may be supplementing folate inadvertently. They give some kind of dog treat, or they give a vitamin supplement and don't think about that they're supplementing folic acid. But certainly many things can cause a decrease of serum cobalamin concentration. And so we certainly have to keep that in mind. But they are certainly clinically useful. They are feasible to perform. In fact, we do them routinely in most dogs and cats that have chronic gastrointestinal sign. We talk about fecal culture, that's not practical, not reliable, not reproducible. Different labs give different results on the same fecal specimen. Serum concentration of unconjugated bile acids. We talked about the fact that in a small number of dogs, this was shown to be useful, but when applied to a larger number of dogs, it really was not reliable. We talked about breath hydrogen being very cumbersome to measure and thus not being reliable. The same for intestinal permeability and mucosal function testing. We need a metabolic cage to collect all the urine, and then measurement is still very cumbersome. Short chain fatty acids in duodenal juice, while theoretically measurable, practically there is no lab offering this for routine diagnostics at the moment. Urine indican concentration, which has been evaluated in people but has not been evaluated in dogs yet. And C13 serum choleoglycine testing, which has not really been in depth investigated in dogs, but would be a theoretical tool, which leaves really sequencing and the dysbiosis index as the only practical and the only feasible diagnostic tool for evaluating the intestinal microbiome. And with this, I will pass on to my colleague Jan Suchodolski, who is going to talk about the details of sequencing and the dysbiosis index. Thank you very much. I'll look forward to some questions. Thank you.
So thank you. So I'm going to continue with about analysis of the gut microbiome and mostly really focus on the molecular parts, in addition what Dr. Steiner already said. So again, I'm an employee of the GI lab at Texas A&M University, and we are providing testing as a fee for service. So that's a natural conflict of interest. I'm also funded by some companies, but generally, mostly of here, what we're talking about is really diagnostics that affect the Texas A&M University GI lab. So we already heard many times that the microbiome is really important. The microbiome is really a part of the whole system. So we have microbes in the lumen of the GI tract, and what they're doing is they are taking some compounds that enter the GI tract. Those could be like dietary macronutrients like protein, carbohydrates, etc., fats, or medications, for example. They're going to take those, they're going to convert them into metabolites. And ultimately, those metabolites itself, they're going to be absorbed along the entire GI tract. And depending on the composition of the gut microbiota, it is really, there can be pro-inflammatory or anti-inflammatory signaling cascades. But I think also understanding that's for diagnosis of dysbiosis, interpretation of dysbiosis is important, is that the dysbiosis itself, changing the gut microbiome, can be, again, induced through medication, can be used to dietary changes, but also what happens on the surface of the host itself is the inflammation, the motility changes, that is also going to change the gut microbiome. So again, we need to understand that both happening together and by itself. So in understanding that we can identify dysbiosis is crucial, but we also have to put dysbiosis in the context of the rest of the presentation of the patients. And furthermore, it's also important to understand that obviously, even if we look at what dysbiosis is, I think we have decent markers nowadays. We have learned a lot the last 5 to 10 years. But again, the GI tract is a very long organ. The microbial function is also very different between small intestine, large intestine, and so on. So we're never going to have one single test really that allows the proper assessment of the microbiome function. So we can see there are changes, but we need to learn with time that what those changes mean and other functional changes and so on. So again, we need to, obviously, limitation is that if we look at a fecal sample or serum sample, it is just one part of assessing a very difficult organ system. Obviously, there are differences in the small and large intestine bacterial populations. I think we have data that suggests if there are changes in the small intestine, for example, through omeprazole treatment or through antibiotics or even small intestinal disease, to some degree, we can actually detect those changes in feces, but it's not going to be perfect. So yes, theoretically, we should see them, but it's probably not in every single patient, or the sensitivity is going to be lower. So again, when you look at for research purposes, really understanding what happens in the gut, a combination approach is really preferred. We want to understand which taxa are altered. We understand, are there some bacterial genes altered, because the functional aspect of the microbiome is important. We can measure metabolomics. That's an upcoming area. So we can measure, for example, bile acids or other compounds that potentially indicate if the function of the gut's microbiome is changed. And obviously, we also sometimes, in some situations, also important to see the localization of bacteria. Are they mucosa adherent in larger proportion than normal, or are they even translocating, like in boxer dogs with Crohn's colitis? So again, different ways of approaching with molecular methods how we understand the gut's microbiome. And then most importantly is actually that we're still discovering that bacteria present everywhere. So this is actually a study from one of our, from Paula Guerretta, who's now a pathologist in Brazil, but she was a PhD student and resident in the GI lab. And what she actually discovered very nicely, so those are actually in the crypts of the colonic crypts of healthy dogs. If you look at H&E stain, you see a little bit of a mucus blob here. If we stain this with silver stain, we can actually see the bacteria present in those crypts. So completely healthy, normal dogs. And we use fluorescence in situ hybridization. We can actually see that those crypts are really loaded with bacteria, in this case, a Helicobacter. So again, we're discovering that actually things that we don't, when we use the right methods, we overlooked for the longest time. Obviously, we look at H&E stains, and now we really appreciate how many bacteria are present in healthy dogs. It's completely normal flora. And importantly, they surprisingly actually are decreased in disease. Again, showing that microbiome, there has some functions, there are probably some immune functions that keep the immune system properly stimulated. They're important. So we need to also better describe, understand the normal bacteria are normal in many situations. Just seeing something on a biopsy does not indicate immediately that is abnormal. We have to really calibrate this against clinical representation and clinical knowledge and so on. So I'm going to talk three major areas. One of them is brief metagenomics, DNA shotgun sequencing, then 16S RNA gene sequencing, and quantitative PCR as one example is this dysbiosis index. Those are the really major molecular tools that are now used, mostly for research setting, but also offered for some individual patients. For those who are really interested, there are good review articles available. That's what published, I think, in 2019 from Marcio Costa and Scott Weiss. And I also published recently a review article that really highlights more the pros and cons of those different methods. So metagenomics is actually one of those methods that has been tracking more and more gain. And I think in literature, there's a little bit confusion. A lot of people call metagenomics as a general term, and they mean generally sequencing. But metagenomics actually should be reserved for so-called DNA shotgun sequencing. And the reason for that is because DNA shotgun sequencing doesn't have a PCR amplification, but rather we extract DNA and then sequence randomly those DNA pieces. And the advantage of that is really that we're not just getting information about bacterial taxa, like for example, the 16S gene, we also, because we're sequencing so many different genes, we could also get the content of them. So it means we have identification of bacteria, but we can also get information about other genes that are present in the sample. And that can be important because function is important, not only who is there, but ultimately what they're doing. So again, the process is PCR dependent. There's no bias, there's no primer bias. So we also have probably a better representation of the true proportions of which bacteria are present in a sample. And more importantly, with those metagenomics, this DNA shotgun sequencing, since we don't really amplify only one gene, we amplify many genes, we can put them all together and have a better resolution at a species and strain level. So that is really important. The cost why it's not really commonly used is because it's pretty high. In order to sequence all of this information, we have to sequence pretty deep, meaning we have to spend more money to get more information and also bigger commercial efforts. Another really concern, I'm going to come back to this quite several times, is that many of those sequencing approaches, we really sequence, when we sequence, data is reported as relative proportions. So we see always as percentage of something. And that is a little bit of a problem sometimes, because obviously, if we report only how something within changes, we don't know if there's an absolute quantification change. And sometimes it's more important, maybe, if a bacterial species or taxon increased much in proportion, not just in the proportions, but in the total abundance and so on. So all the sequencing methods is important to understand. They really report only relative proportions. I think 16S RNA gene sequence, that is most commonly used way of assessing the gut microbiome in the research studies, but nowadays there's also some laboratories who offer this technique for individual patients. The reason why the 16S RNA gene sequencing technique is really so favorable, because it's pretty cheap, relatively cheap. And what it allows us to do, we extract DNA, then we amplify this one single gene and it can sequence it. So with one PCR, we theoretically could describe all the different bacteria that are present in a sample. So we can really identify many different species and so on. It helps us really to identify changes in bacterial communities. However, because we amplify only one gene, the resolution sometimes is very low, meaning we can go down pretty on a genus level, but often we cannot distinguish, for example, an E. coli from a Shigella from a Salmonella. On this level, we cannot really, they're too similar. So we cannot really say this is an E. coli or is this a Salmonella, for example. So that is a problem. Again, as I said before, all sequencing-based techniques really report only relative proportions. So again, no absolute quantification, which often for research studies can be sufficient, but sometimes for patients and so on may not be really enough. Another really major area is, and I think that is really important to highlight, there's a really low reproducibility between runs, because it's a so-called an untargeted assay. It's not reproducible. So in our lab, for example, when we do research studies, we do not take data from one sequencing run and compare to data from other sequencing run, because we discovered there's way too much variation. So ideally, you have to sequence everything in yourself. And that makes it really a problem to compare, especially when it comes to individual patients over time. So it's for me, very, very difficult to see if that is really reproducible. Then again, when you really look very critical literature, there was not one single publication that truly showed a true analytical validation of amplicon sequencing. So that means we can really use it for research tools, but I would be very careful to look at this and how reproducible is it really for one patient if you want to follow him up over time. And the second problem is also that's not really a problem, it's actually a good way for research areas, is that technology changes very rapidly. That means machines getting changed, the DNA extraction methods, all of this changes very rapidly in a good way. So we get better information, but because of that, it's also very difficult to maintain this, the data in a how to call it, an assay to be really reproducible. So again, as a research tool, great, we can really get a lot of good information, but because of all those changes in technology gonna affect the results, it's not possible probably to compare results today to results that I gonna generate in 1 year and so on. And again, that is really important to highlight, that it's a not reproducible technique. But generally, when you look at all those sequencing techniques, what they do is, so we generate those sequences, they get processed, quality control, and they get identified against those databases. And databases help afterwards to say, well, those sequences are, for example, E. coli, those sequences, and so on. And again, this really is two problems here again. Great for research tool, but again, all of those changes, the database change over time. That's why all my data, every single time I run it, may slightly change. So it's nice to compare within a study, but again, always be careful comparing results across papers. And that's really also a little bit a problem. When we read all those microbiome papers, everybody reports a slightly different. Somebody who is really versed in the microbiome, we can make sense out of it. But again, it's very difficult to really compare those microbiome reports across many different studies. But generally, we report 3 different things. The taxonomy, that means which bacterial taxa are present. Then diversity, there's a beta diversity that really shows how much is the community in one sample different from the community in another different sample. And so-called alpha diversity, how many different species are in one sample. So taxonomy is important. So again, which bacteria are present? And as you can see, again, I'm highlighting this because it's always relative proportion. So usually we always report them as a percentage. How much percentage is there? Important to understand is also there's a lot of range of normal. For many taxa, we know that there's a huge range of normal. So for example, this is completely healthy dogs. Some dogs have, like one dog here has 50% Bacteroides, another has almost zero. That's also, so some of those is very important. That's why looking at one single bacterial group is really difficult to say it's normal, abnormal. That's why we always combine the data and try to put this all in a complete way. Then alpha diversity, again, that's the number of bacterial species in each sample. And this is something that actually is often, often discussed as if you have a higher diversity, so a higher number of species, that would suggest a more stable microbiome. And that's probably true, and it makes sense. And if an ecosystem is highly diverse, there's probably a lot of different functions and stability and so on. So it's probably more healthier. And so, but again, we have to be a little bit careful here because as you can see, this is a study from horses, but we see this also in other different dogs and cats, is that here, for example, healthy horses on different diets, dietary changes, some changes in antibiotic-associated diarrhea, salmonella. Yes, there is statistically often a lower diversity, but there's a huge overlap between healthy and disease. So again, diversity by itself is a concept that is probably true, but we cannot really predict from one single sample or from one individual the diversity, if that's a good or bad microbiome. And then we're looking at so-called beta diversity. And beta diversity means is how does one sample really differ in the composition from a different sample? And so what that means is, for example, we're looking at really the distance from two different dots. So this dot here, if it's closer to this dot, so they are more similar. If it's far away from another dot, they are very different composition of the gut microbiota. So this is a nice way of showing other difference in gut microbiota. But again, the problem a little bit in literature is, and also for individual patients, is that when we do this, it's difficult to understand what is the normal. Okay, so yes, here we can see that diet E seems to be different from all the other diets, diet A through D. But the problem is because we're just comparing them to each other, we're not comparing them a huge reference population. So it's difficult to see, is this effect really large or is this effect rather small? So that is also a problem when we look at all those microbiome data. There's a lot of statistical reporting saying, okay, there's something different about it. However, it's difficult to judge what is the magnitude of it. And I think that is really the problem. There's no reference intervals that we can compare it to. So it's a lot of speculation about the size effect. And that's why in literature, we hear this a lot. This changes something statistically, yes. But the question is, how big is the effect? Sequencing tools, because they don't have really reference intervals, cannot really tell us that. And so the other option to really look better on gut microbiome is actually quantitative PCR. And the advantage of a quantitative PCR is that it can be very reproducible. It measures the abundance, so we can have standard curves, we can measure it. We can really, because it's a simple assay, we're always running one assay after another assay, we can really target specific either taxa of interest or functional genes of interest. And if we really use the same DNA extraction protocol and the PCR protocol, it is actually a highly reproducible test. And because it's reproducible, for those targets, we can really compare across studies, but we can actually, and I'm going to show you this a little later, is we can truly establish reference intervals for some taxa of interest. So we can do that. The disadvantage a little bit is that obviously we can only do one assay after one assay. That means we usually have a limited number of targets. So we cannot look at all like 50 or 100 bacterial taxa that are part of the microbiome. But usually it's a very highly reproducible assay, and it really allows us to establish reference intervals. And so I think that probably the best example is the dysbiosis index that we kind of started or published now in 2017. And how we did this was actually really focusing on some bacterial taxa that we know across many different studies. Some of those bacterial taxa usually decreased, some of those increased usually. So we targeted those ones with a real-time PCR. And then with time, we also understanding that many of those taxa have important functions, like Faecalibacterium is a really very beneficial bacterium that has anti-inflammatory secretions, produces short-chain fatty acids. Fusobacterium in dogs is also very beneficial, and Clostridium hydrononis, this is really what seems to be one of the key species because it converts bile acids, primary and secondary bile acids, and through this function, it has really major implication on the homeostasis of the gut microbiome. So the nice thing about this is that over time, we're able to actually develop reference intervals in healthy dogs, and we validate them across many different clinical studies. So I think we have a pretty good understanding is where most healthy dogs fall for those groups. And we can also, because I said before, looking just at one single group may not be sufficient. There's a huge range of normal, but if you combine all of those results into one single number, we get a so-called dysbiosis index. So this number here, for example, is the combination of the abundances of all those 7 groups together and how they change. And so we define at the moment below 0 is a normal dysbiosis index. So most healthy dogs fall in this range. However, important is that some dogs may have some of those bacterial taxa out of reference intervals. So for example, here in blue, if one of them is abnormal and so on, and you can see the more abnormalities we have, the more it increases. And so it's important to understand that even if a normal dysbiosis index, if there's already some changes in gut bacteria, in some of those, we call this a mild dysbiosis, a minor dysbiosis, 0 to 2 moderate shift, and above 2, which is pretty specific for dysbiosis, I believe. It is a major shift. And we're going to discover more and more that actually the shift is important. The shift is important. We have studies that shows that the higher the index, especially this 7, 8, 9, or 10 range, those are usually dogs that either don't even respond to FMT or there's something really major wrong in the GI tract. Because I think what we're also discovering more is that the gut microbiome is really mostly a marker, a reflection of an optimal gut environment and of severe changes. So the higher the index, it suggests the more severe changes within the GI tract. And we can see this in the correlation with the dysbiosis index with full 6NS sequencing. So again, in the dysbiosis index, we're really measuring only a few PCRs, but they actually predict pretty well how diversity is. So again, you can see here a very high dysbiosis, the higher the dysbiosis index, 5 to 10, also the lower the diversity. And again, diversity, while it's a little concept, it still shows that in many studies, if you have low diversity, or in this case, high dysbiosis index, it would suggest a really poor response to therapy. We also discovered that some bacteria that also measured by qPCR, so Faecalibacterium, Fusobacterium, the more we have of those, so again, those are beneficial bacteria in dogs, the more we have of those, we have also a high diversity, again, potentially a more indication of a healthy gut. So again, the dysbiosis index really correlates pretty well with the diversity of species richness in 6NS sequencing data. We also have shown nicely that the index also shows how the better diversity is. So as you can see here in yellow, dogs with a high dysbiosis index clearly cluster away also on the 6NS data, so on the full sequencing data from dogs with a low diversity. Again, so the dysbiosis index is a good proxy if the microbiome shifts. But very intriguingly, though, is that we discovered that Clostridium hydrononis, this bile acid converter, is really an important bacterium. Because as you can see, low Clostridium hydrononis, so dogs that don't have low Clostridium, again, also cluster really from dogs that have a high. So again, we can separate the microbiome based on the presence or absence of Clostridium hydrononis, suggesting that it's an important bacterium. So if it's there, we have a completely different microbiome composition than if it's gone and so on. And we also see this in the dysbiosis index. So as you can see, those dogs in red here, they have a decreased abundance of Clostridium hydrononis. So again, if we do not have Clostridium hydrononis, that really by itself indicates pretty clearly that there is a shift in the total gut microbiota. And the reason for that is really that Clostridium hydrononis, again, it is this important bile acid converter. So it converts primary to secondary bile acids in the canine intestine, to some degree also in the feline. We're learning more about it. So that means if we do not have hydronosis, we're also not going to have conversion. We're not going to have secondary bile acids. And the secondary bile acids, as I mentioned before, and I'm going to mention later on, are important in many different pathways. So that by itself is a crucial pathway that also keeps for systemic health, but also for homeostasis of gut microbiota. And again, why is this pathway so important? Because again, from the liver, we have primary bile acids. They get secreted after a fat meal into the GI tract, and the gut microbiota convert them in secondary bile acids. And I said before, in dogs, it really, Clostridium hydrononis is the major one. So we need this bacterium to convert this. And again, it's an important pathway because if this ratio between primary to secondary bile acids is correct, we're going to have really proper signaling. to the liver. We're going to have anti-inflammatory signaling cascades to other organ systems. We're going to control pathogens. We need those secondary bile acids to inhibit the growth of some Clostridial species like Clostridium difficile or Clostridium perfringens and so on. So again, this pathway via Clostridium hydrononis seems to be really, really a key pathway in the microbiota of dogs. And we can also see nicely how really over time we discovered that those reference intervals are pretty important, because it shows us that when you, for example, when you look at healthy puppies or puppies that are just born, usually we're born pretty sterile, but immediately after birth, the GI tract gets colonized in the beginning from environmental bacteria, like for example, E. coli and so on. And just really over time, over a few months, when the normal digestion starts in, so they get transferred to regular diets, such as enzymes, pH changes, all of this normalizes or becomes like an adult, the microbiome really normalizes. So we can see that over time, we have a lot of E. coli. This is completely normal in healthy puppies because they are the first colonizers. Over time, they decrease, come into the reference intervals like we see in adult dogs. Fecalibacterium, those beneficial bacteria, also very low in the beginning, in the first 4 months of life. They increase and then come in the reference interval of normal population. And that's why also the dysbiosis index, because it's a combination of all those groups, is increased because we have a lot of E. coli and low of, for example, of those anaerobes. But again, after the period, somewhere between 3-4 months, it normalizes, and really afterwards is really in the reference interval, what we usually described for healthy dogs. And again, understanding all those combinations is important because that we also don't over conclude if we see potential enteropathogens. So again, that was actually a very surprising finding that a lot of puppies have, when they are in the younger age, completely healthy, no signs of diarrhea, but they have increased Clostridium difficile. But you can see that suddenly week 7 to 9, without any major intervention, difficile is actually decreasing. And why is that? Because again, we notice really strong association between the normal flora, Clostridium hydrononis. So as you can see, Clostridium hydrononis really starts appearing at 7 to 9 weeks. And we know that Clostridium hydrononis is an important converter of bile acids. So as you can see, it really coincides very strongly with appearance of secondary bile acids. And there's very good data out there that this pathway is really important in reducing some of those enteropathogens. So again, what we're discovering more and more, Clostridium difficile and Clostridium perfringens are usually overgrowth bacteria. They're probably not really that major in dogs and cats. And if they are overgrown, it's probably because we have lack of this pathway. We have lack of Clostridium hydrononis and also lack of conversion of secondary bile acids. The other really important reason is why we have those markers now, for example, a really validated assay that we know over many different studies that it can really show us what the microbiome is. That also helps us to put this in perspective, changes that we observe in some studies. So remember, for example, I showed you many studies report something and they compare only one to another. So here in this study, so we have there's a control versus the test, and you can see perfect separation. They're very completely separated. But again, the problem with that is it doesn't really tell us how big is this effect. Is it really completely abnormal, or is this all still within all the normal dogs that are out there? Because again, we don't have a reference interval. If we put the same samples in the dysbiosis index, yes, we see there's a shift. The dysbiosis gets lower, but again, as you can see, the shift is probably not that major because it's still within the reference population that is established. So again, that really helps us also put all those microbiome changes in an important context. We can also use validated assays that we can see, does it actually change before and after treatment? I think FMT is a perfect example. If we have an increased dysbiosis index, so unfortunately in this dog, for example, it's an increased dysbiosis index. He doesn't have Clostridium hydrononis. Because of that, he doesn't have conversion of secondary bile acids. After FMT, 1 week, we can see that the index normalized. Clostridium hydrononis went in the reference interval. And we also have the conversion of bile acids. And we can also see that after 4 weeks, the microbiome is still normal. And this is kind of important because we're learning more and more FMT. We're going to talk about this in one of the last talks together with Dr. Torresen, is that we can, to some degree, how the microbiome is in the beginning, potentially, we still have early times, but also in human medicine, there's more and more coming out that it can, to some degree, be a predictor of the response to FMT in general therapy. Because again, the microbiome can be used as a marker of how abnormal the gut environment is and so on. Interesting is also that with time, we're discovering too that often we think, well, dysbiosis equals diarrhea or vice versa. No, it's not. Diarrhea is not necessarily always associated with dysbiosis. It often is, but it's not the same. And interestingly, the dysbiosis pattern vary between different phenotypes. Okay, so yes, dogs with chronic diarrhea, chronic enteropathy, many of them have dysbiosis, also EPI and so on. So we're discovering dysbiosis is not something that we should immediately address with antibiotics, because also many dogs that are food responsive may have dysbiosis or idiopathic IBD. But interesting is that in chronic enteropathy, most, I would say 50 to 60% of dogs with chronic enteropathy have a decreased abundance of hyalurones. And this doesn't really happen in acute diarrhea. Very few dogs with acute diarrhea have decreased hyalurones. Even in dogs with acute hemorrhagic diarrhea, the majority of dogs has hyalurones. So again, suggesting that hyalurones is really a group that, if in chronic GI disease, there must be a chronic process. And it kind of makes sense because, again, it reacts to the gut environment. So with time, with a chronic process, there are more changes, and the mucosal level. So that's more an indicator that there's actually some chronic process going. However, we know, though, that there are some treatments that we need to always put in the context. We know that antibiotics definitely can change the gut microbiome, can also decrease Clostridium hyaluronis. So ideally, depending on the antibiotics, and unfortunately, there's also a very huge individuality between patients, but at least 2, ideally 4 weeks of antibiotics. We also know that omeprazole can induce a dysbiosis. So again, this is highly reversible, and it should be really reversed, at least usually typically resolved between 10 to 14 days after the end of omeprazole. But again, what's interesting is that omeprazole, we also, again, as a saying, we can identify different dysbiosis pattern. So for example, this is healthy dogs on omeprazole. Again, all healthy within the reference interval. On omeprazole, we see an increase in dysbiosis index. When we stop omeprazole, they go back. But what's kind of very interesting is that in omeprazole treatment, it's really lactic acid bacteria that increase, so Streptococcus goes up. And the reason actually is kind of makes sense because we did studies like in 2010 where we showed, and we looked fish in the stomach and the small intestine. When we put dogs on omeprazole, we saw that obviously the pH, in the beginning, you have mostly Helicobacter in the stomach. If we increase the pH from 2 to roughly 4, the first bacteria that can really grow in this environment is lactic acid bacteria. For them, they can survive at this pH, but others cannot yet. So they have a growth advantage. So that's why we have this increase in Streptococcus. It makes perfect sense. And again, saying that dysbiosis is not dysbiosis, because on omeprazole, those are healthy dogs. There's no reason they should not have hyalurones. So again, omeprazole increases Streptococcus. That's why the dysbiosis index, but Clostridium hyaluronis is in the normal range because those are all healthy dogs still. So again, we're learning more and more that there's different dysbiosis pattern out there. Different story with antibiotics, broad spectrum antibiotics like tylosin, metronidazole, they truly have a profound effect on the dysbiosis index. The return is very individual. Some dogs can return within 2 weeks, some dogs within 1 month, but there's always a few dogs that might even need longer. And especially Clostridium hyaluronis is truly very sensitive. So this key bacterial species is very sensitive to antibiotics. So again, we always need to have a good antibiotic history for the interpretation of Clostridium hyaluronis. And the other really important aspect where we're really also learning more and more is that, again, dysbiosis is often a marker of an abnormal gut environment. We know that in a chronic GI disease case, once it's chronic, there's a lot of abnormalities. I mean, we did a lot of metabolic functions, so functional metabolomics, and we discovered that many of those markers are really truly abnormal in the GI tract. So that means because there's a lot of changes. We have a decreased mucus layer, we have changes or the destruction of the epithelium. Because of that, we have also decreased the brush border enzymes, so meaning absorption. So a lot of compounds that should be absorbed in the small intestine are not absorbed. They are available for bacteria and they overgrow. So we have this bacterial dysbiosis. So dysbiosis is often really a reflection of the gut environment and so on. And we discovered that actually, especially in chronic enteropathy, it also takes a long time. So there's a lot of studies out there that look at, like follow-up of microbiome changes, 3 weeks, 8 weeks. So we have really published a paper recently. This paper really is only a small number of dogs because obviously we're just discovering, but we have lot of data out there that is unpublished yet, but really has the same trends, that it takes a long time for the gut's microbiome to come back. But generally here in this study, some of those bacterial tags like Fusobacterium, abnormal baseline in those 5 dogs, really there's a trend slowly, but the real, after more than 1 year, then slowly comes together. Because again, it probably takes a long time for the gut microbiome to normalize, because all those changes that are present are probably still there and they take a long time to improve. So again, I think it depends on the purpose we want. There's definitely a complete assessment of the microbiome requires computer methods. They're obviously only available for research settings, but I think what we're discovering more and more, that again, like in every other organ system, we can focus on some of those most important pathways, and we can correlate them to disease, correlate them to different outcome measures. And really, I think the future is going to be that hopefully with a few selected markers, we can still pretty well describe what happens in the gut and how normal or abnormal it is. I think important for me to highlight is that the dysbiosis needs to always be evaluated in the context of disease. I think in the past, we always said dysbiosis is something that we should treat by itself. No, I think dysbiosis really reflects the gut environment. So often dysbiosis is an early marker. We see this because it reacts to the gut environments. We can potentially, in some cases, could use it as an outcome measure. Does the gut environment improve? But those are probably more studies needed, especially in FMT, but also over time in chronic enteropathy. And again, we have initial assays available. The dysbiosis index, if it's validated, we have more than 50 different publications about it. So we have a good feeling that where, for some groups, normal is. That means it's reproducible. We have reference intervals. We see that in increased dysbiosis index with low Clostridium anonis, which is the keystone species, often in chronic enteropathies, if we exclude antibiotic usage. We can use those markers to really screen FMT donors, especially Clostridium anonis. I mean, we see that roughly 10 to 15% of clinically healthy dogs don't have this bacterium. Not sure why. It could be that they have antibiotic history and never recovered, and like some dogs do. There could be some underlying subclinical already changed in the gut environment to discover. But again, it shows us that we identify some important groups, and I think ideally for donor screening, those are important. And also initial data, again, more data is needed, but again, it appears if you have very high dysbiosis index, it's really low microbiome diversity, which potentially reflects a more severe change inside the guts. And that possibly is a negative predictor for FMT response. We have small data sets in humans, but actually in dogs, but actually when you read this in humans, this diversity comes often up. The diversity low is a negative predictor, high diversity is a better predictor for outcome measures and so on. So again, with this, I want to acknowledge a lot of my collaborators that are really needed for really do all those studies together, because the more we look from different angles, from healthy to disease phenotypes, the more we can learn. A lot of grant supports. And with this, I'm happy to answer any questions.
Hello, and welcome to the questions and answers session of the Purina Institute Microbiome Forum, putting microbiome science into clinical practice. I'm sure you had 2.5 hours of wonderful learning and lectures, and now you have time to ask your questions from the eminent faculty that share with you their experience and knowledge. We received a lot of questions you submitted in the questions and answers session, and you can continue to do so. And without any delay, I'm asking Dr. Jan Suchodolski to moderate discussion and invite all the speakers to participate in that. Let's enjoy it.
I was muted. Thanks, Natalia. Yes, I would definitely echo that we had a really, thank you for everybody presenting. We have a lot of questions, so it shows there's a lot of interest in the microbiome, and especially clinical applications. So we really want to take our time and extend questions to all our speakers. And I wish that we got all nice discussions and really use some practical findings or new experiences. So let me start. There's a lot of antibiotic questions. So let me start with one. Can the microbiome be restored by nutritional support in developing puppies exposed to antibiotics? So maybe that's a question that I think clinically we don't know much yet, but maybe Gail, in some of your studies, do you have any experience? Can we restore the microbiome in developing puppies?
Oh, it's a great question. And like you said, there's a lot of room for more research in this area. I know we have data showing microbiome over time in puppies and showing how it's affected by weaning stress. And I know that we can affect that with probiotics and prebiotics. We haven't done studies with antibiotics per se. Mm-hmm. So maybe Linda, Mike has, oh, I'm sorry. I'm passing this on to somebody else.
Okay. Mike, do you have any experiences?
Yeah, I'd like to echo that's really a great question, that we do need a lot more data. You know, we believe that we can induce some clinical improvement, but whether or not the microbiome recovery correlates with that clinical improvement is still where we're lacking a lot of information.
Mm-hmm. I know, Stefan, and you have some experiences FMT in your clinical practice in Munich. This is also with young puppies too?
Actually, I think it's a very critical phase if you have a young individual and a puppy. So I think also it might be very similar to human medicine, that they might progress to chronic disease. So what we see frequently is in young dogs treated, for example, with metronidazole. And metronidazole might not be the big problem in adult dogs, but I think in young dogs, maybe you really can induce a significant dysbiosis and maybe chronic disease. And in these cases, we really sometimes consider to give fecal transplants.
Okay. So in line with that, do also injectable antibiotics have any or as much impact on the microbiome as parentally, or I guess orally administered medications? This question was open to anybody. I can give a little bit of background. So yes, we have a few studies. Actually, we have really studies. Most studies that we did where we looked really at the microbiome with oral antibiotics. But we have a few cases that we know also after injectables, there's also a huge dysbiosis can be induced. And the reason is that most antibiotics ultimately going to reach the small intestine to the liver, to the bile and so on, that can also induce a dysbiosis. So yes. Dr. Gale, have you ever found that different strains of probiotics have an antagonistic effect on each other? In other words, is it best to supplement with few but effective strains rather than several strains?
That's a good question. I think it depends on what the strains are. I don't know that we've seen that if you take two strains together, depending on the strains, you can blunt the effect. I don't know if I've ever seen it totally disappear, but I think it just depends on the product. I mean, there's a number of good multi-strain products that are on the market that are efficacious. So it's unfortunately just one of those. It depends on what the strains are. Answers.
There was a lot of questions about dietary influence on the microbiome, especially dysbiosis index. So one question was, now those are directed to me, have you ever looked at changes in the dysbiosis index with various diet compositions and types? And there's actually a follow-up question which follows that is, how do we determine what is and what is not dysbiosis, given the differences in microbiota in dogs, cats, fed kibbles versus canned diets versus raw foods? And I think that's a great question. And I think as we're learning with any other, this is my personal opinion now, and based on, I think a lot of data that we have is that I think there's obviously a huge range of normal, what is normal. However, we see very clearly that most diets, yes, when you look at just the microbiome itself or the dysbiosis index, don't make any changes in that. And again, because even if you know that there's plenty of studies out there that have shown that if you give fiber, for example, yes, a few groups change, but the magnitude of change is rather minor, especially when you compare this to antibiotic usage or GI disease, where we have a much, much bigger shift and larger shift in that. So again, I think we're learning definitely diet has influenced fibers on the micrometabolome, but it doesn't necessarily lead to a very big shift. And I think like also probably one problem with microbiome studies is that most papers have really compared one small thing to another small thing and never to the big reference population. And so now, since we have now markers that kind of define a reference population, we truly see that actually the influence is not as large as we thought it is. And I think so. So generally, the dysbiosis index is not affected by those components. So there's much more increased antibiotic usage and in GI disease. A question for Linda. As a general practitioner presented with a dog or cat with diarrhea, what do you recommend as the primary workup and management of the case? And what would be the subsequent step if the previous one wasn't effective? So I guess, what are your steps in the workout?
It depends on the age of the animal and how, if I find any markers of severe disease, such as a low albumin, a low cobalamin, high disease activity. That's been shown in dogs, but the negative prognostic markers in cats are not as well defined. But look for severe disease, such as if the animal has lost a lot of weight, has a lot of diarrhea, or has completely stopped eating, or things like that. Then you need to be a bit quicker in your workup. But if you don't have markers of severe disease, which you know when you have done your basic workup, so blood works, and then I mean what is often called as a full profile or total profile. And if they've had problems for a long time, I always check cobalamin and folate. TLI if it's a dog losing weight and has diarrhea. TLI in cats if the cobalamin is low. Otherwise, it's usually not worth that. And PLI in cats if they are anorectic and have been vomiting on and off for a long time. And I always check for parasites, the first thing I do, especially in young animals or dogs used for hunting or cats that are out hunting for prey. Then your likelihood of finding parasites is much higher. So the basic workup is checking for parasites, doing the blood works, and also ultrasound of the abdomen is one of the first things we do if the animals has had clinical signs for a long time. But in puppies, we often just start with a parasite test, including Giardia, because you often find the answer there. And if you don't have any evidence of severe disease or a marker for negative prognosis, then I start with a dietary trial. Often I go for hydrolyzed diets if they haven't tried that before, or if they are severely anorectic, we usually do a test trial. So we have open cans of kibbles. So we can actually serve kibbles to the dog or the cat, and sometimes that works for cats too, so they can choose what they find most interesting. And that's often a good idea, because then the owners are much more positive about buying that type of food. They often say that, oh, it's not going to work, my dog is not going to eat that. But if you actually serve different types of kibbles, then usually they show interest in one of them, and you can often send them home with that type of diet. And I often use a probiotic that is proven for chronic enteropathies in dogs. The same type of probiotic hasn't been proven for chronic enteropathies in cats, but it has been shown that in chronic constipation, it will improve the microbiota and also decrease inflammation. So you can say there's some evidence in the pilot study in cats too. So if I want to speed up the process, I start with a new type of diet and the probiotic, and then we go from there.
And if that doesn't work, what will be your next step?
It depends on how desperate the owner is and how the animal is doing. We could do another dietary trial. We sometimes try home-cooked diets, mostly for dogs. There are good basic hypoallergenic diets in clinical nutrition. So there are some recipes you could use there, or you can talk to a nutritionist about it, because sometimes the home-cooked diets work better than the hydrolyzed diets or the novel protein diets. If they don't want to go for another test trial, then we usually go for endoscopy and start some type of anti-inflammatory treatment with corticosteroids or cyclosporin or something similar. Unless the animal has diabetes, then you have to avoid the corticosteroids, etc. But I always try with a diet before I go for endoscopy, unless it is a dog with really low albumin that don't respond to a low-fat diet, and we want to rule out lymphoma. So we don't wait very long with those. But that's why I want to know if you have signs of severe disease or not.
Great. That brings me kind of to the next question that is quite often answered about owners want a quick fix with diarrhea and antibiotics and so on. So how do you see antibiotics in this workup? So maybe Linda first, and then Stefan, and then Jörg.
Yeah, we don't do the typical workup of antibiotic trials. So classically, you start with diet, and then if the dog don't respond to the diet, you go for antibiotics for 2 weeks. And if the dog responds to antibiotics, you call it antibiotic responsive diarrhea. And if that doesn't work, you go for corticosteroids or other types of anti-inflammatory treatments. And in Sweden, we have quite strict rules regarding antibiotics. So I haven't used antibiotic trials for, I would say, 10, 15 years. And there are so many dogs that do respond to antibiotics that also respond to other types of treatment. There are some dogs that truly don't respond to anything else than antibiotics, but there are very few if you skip that step. So the quick fix is to avoid the antibiotics if you have because you asked something about the quick fix too, didn't you? Yeah. So yeah, there are actually 2 studies that both were published in 2019 looking at metronidazole in acute diarrhea. So in the first, that was Langlois, published in JVIM in 2019. They had 31 dogs with acute diarrhea. They gave them a placebo or metronidazole. And the metronidazole, well, those dogs, the diarrhea stopped after 2.1 days, and with the placebo, 3.6 days. So the difference was extremely short. And that doesn't really justify to give a broad spectrum antibiotics that will kill a lot of the beneficial microbes just because you have 1.5 day less of diarrhea. And in the other study by Schmalberg, they had 60 dogs, and there was no difference in the length of diarrhea if the animal was given a probiotic, metronidazole, or placebo. So actually metronidazole is not that much of a quick fix that we often think as a clinician. So the quick fix for me would be to send the animal home with acute diarrhea with one of those clay preparations that usually contain some prebiotics as well as a low dose of probiotics, because they usually respond to that, most of them.
Mm-hmm. Stefan?
Okay, so yeah, maybe if we talk about acute diarrhea, I have the same opinion as Linda. And but I think it's more that we have this feeling of tradition to giving antibiotics, because maybe 15 years ago, when I was first in private practice, it was completely normal to give antibiotics to every dog. And so I think clinicians are very used to give antibiotics, but nobody was considering not to giving it or comparing it to placebo, for example. And now we have these studies showing that nearly every dog with uncomplicated, that means a stable dog without fever and all these inflammatory signs, an uncomplicated diarrhea case, they usually respond or recover in 4 days. And sometimes it lasts 7 days, but usually in 4 days they are happy again. So that means usually we do not need antibiotics. And I completely agree to Linda that we create maybe a dysbiosis by giving antibiotics, which we do not want. And so we do not give antibiotics on a routine basis. We always select these cases very thoroughly.
And in chronic diarrhea, what is your workup?
Yeah, in chronic diarrhea, I have also to admit that we treated several years ago nearly every dog first with also uncomplicated chronic diarrhea with diet, and then we had a tylosin trial or antibiotic trial. And in my feeling, we had good responses. And in the literature, we think about 15 to 20% of dogs will respond to antibiotics. And but I think that this is usually not a long-term success, because if you stop antibiotics, usually it comes back. And I think that we more or less treat the immune system, or we help the immune system by giving antibiotics, because we reduce the number of bacteria, we reduce the number of bacteria attached to the mucosa, and this is less stress for the immune system. So for the time, as long as we give antibiotics, everything is fine, but the underlying disease is not treated, and we stop the antibiotics and they will have clinical signs again. Sometimes we are lucky and we give antibiotics for a couple of weeks, and also the underlying disease might improve and they will be stable without antibiotics. But usually what we know from the literature and from our cases is that the diarrhea comes back when we stop the antibiotics.
So at which stage of your workup in chronic patients are you using antibiotics now?
Actually, I changed my opinion, because a few years ago, I always treated first with antibiotics and then with steroids. So we do the same workup as Linda, that we rule out parasites, do diet trials. We usually perform 3 or at least 2 to 3 diet trials, because I think that's very important. We modulate the intestinal microbiota by probiotics, sometimes by fecal transplants. And if they're not improving, most of the time we recommend to take biopsies. If we see a significant inflammation, that means a moderate to severe inflammation, and from the cell type, it looks like an immune-mediated inflammation, that means lymphocytes and plasma cells, then usually we do a treatment trial with steroids. But if it works, we reduce the steroids very quickly and add another immunosuppressive or anti-inflammatory drug, so that we do not see so many side effects. If we do not see a significant inflammation on the biopsies, or if they are not really responding well on the steroids, then sometimes in these selected cases, we try to give antibiotics and see the response. But these are really a few selected cases at the moment. And I think the same is in dermatology, for example. Many years ago, every dog with bioderma, they received antibiotics. And nowadays, they also treat underlying diseases. They use topical treatment, and they are not so focused on antibiotics anymore. And I think we can handle this, most of the cases, without antibiotics.
Jörg, what is your thoughts about the workup?
I show my age that I'm a little bit more positive antibiotics for chronic disease. Acute disease, we don't need to talk about it. I mean, Stefan in his studies and others have shown very well that there is absolutely no benefit of antibiotics, and if anything, that's detrimental. For chronic disease, it's a lot more complicated, because I think, on one side, of course, Jan, you've shown that tylosin leads to dysbiosis. But I think Westermark studies are still very powerful, who showed that there was groups of dogs that didn't respond to anything else, that responded to tylosin, and when you took them back off the tylosin, they had a remission. They went out of remission. And so I believe that for my cases, the sort of chronic cases that don't respond to diet, which 50 to 60% of them do respond to diet, so that takes care of a big number of them, and then you take out the parasite ones at the very beginning, even before then, it doesn't leave that many cases. And I think, and I wouldn't call it an antibiotic trial, because I don't use anything but tylosin. So, and we're not really sure, how much of tylosin's action in improving these dogs actually is truly an antibiotic effect. It could be an immune modulating effect. We don't really know what the effect is. But I do believe tylosin has few side effects. Yes, it does cause further dysbiosis from a scientific point of view, but I do see a lot of dogs that respond to it. And if you treat long enough, 6 to 8 weeks, many of them stay in remission. So I know I'm sort of, I'm probably a little bit, stubborn on that. Certainly FMT is a good alternative to that. And I'm recommending that more and more. But again, a lot of that is not known. What in fact happens and why tylosin improves these patients, I have no idea.
Mike, you raised your hand.
Yeah, just a comment back to the metronidazole addiction that many people of my age group had, earlier in our careers. Part of that stemmed, of course, because of the high prevalence in most areas of giardiasis as one of those differentials. And so metronidazole was a go-to. And we've actually, I think I mentioned in my talk, the Fenimore paper, where it's ingrained, like the shelter that we had studied recently for nonspecific small bowel diarrhea, they wouldn't stop giving metronidazole. And so if we wanted to do a study in that shelter, we needed to find a way to gather data without changing their current protocol. And that's one of the Enterococcus faecium strain SF68 studies, where we just did the nonspecific diarrhea dogs with diet change and metronidazole, and then half the group got the probiotic as well. And, I always call it getting off the toilet a little bit faster. We did show an effect of a clinical effect of that treatment. And then just to remind the listeners, Jan's team and Jorg's team, we coupled together to show if you were really going to do a nonspecific diarrhea challenge with antiparasiticide, that actually using benzimidazole makes more sense in these days because Bantel and fenbendazole used for Giardia and then the nematodes don't change the microbiome with the tools that we use in that study, whereas metronidazole obviously does. So back to Linda's chat about, doing the parasite trial. As a parasitologist, I love to do diet plus fenbendazole in the cat or dog, or diet plus Fabantel in the dog as my therapeutic trial with or without probiotic before going on, to the antibiotic trials.
Mm-hmm. Linda, you have one more comment to that?
Yeah, I'm just trying to unmute myself. And of course, I do use antibiotics at some time points, but I think we should save them for the last step. So I think really you should try all the other options first, and also thinking about the One Health perspective. We don't want to create antimicrobial resistance in the pet that can actually spread to the owner. So really, I think we should save the antibiotics as the last step. And then the ones that are truly antibiotic responsive, they won't respond to corticosteroids, but actually quite a few dogs will respond to metronidazole. When you stop the metronidazole, they will get diarrhea again, and some of them will respond to corticosteroids or other immune modulatory treatment or FMT. So my point of view here is trying to save the antibiotics as the last step for the chronic ones.
Stefan, last word to this question, and then we...
Yeah, I just wanted to say that I'm happy that Jörg mentioned that tylosin is working, and I think we need the whole information from different clinicians. There's not one truth answer to it at the moment. But I wanted to stress a little bit that sometimes in Europe, I hear that metronidazole is a very small spectrum antibiotic. And this is true, because if you want to treat a septic patient, it will very likely not work because it works against anaerobes. But from the view of the GI tract, we should know that most of the bacteria are anaerobes. So we have a very, very strong impact on the intestinal microbiota, and we kill a lot of anaerobes. So, and this might be also one reason why this antibiotic is working so well, because we reduce the bacteria. But it does not mean that this is a small spectrum antibiotics and has not a very impact on the body.
Mm-hmm. This is perfect because it answers two more questions. That's, can metronidazole or Tylenol administration lead to a decrease in the good bacteria like Clostridium nonis? I think yes, it does. We know that. That has been nicely shown. So it definitely decreases the number of good bacteria. And the question was, why does metronidazole seems to work so quickly for many dogs with diarrhea? That's a question for Stefan. Is it due to some anti-inflammatory action or an antibiotic effect? I think we answered the question to some degree already. Your thought was that it's decreased the total number of bacteria, and because of that, maybe the immune system responds, but generally it does not really improve the microbial pattern, correct?
I think it would be too simplistic to think that we kill the bad bacteria, the harmful, and we save the beneficial bacteria. I think that's not working. And yeah, so I think that this is less stress response of the immune system to the lower number of bacteria attached to the mucosa. And I think that's the main mechanism.
Great. Let me ask a question to Holly. I think it's a great question about low diversity in bats. I think very actual question. Is it related to the high prevalence of viruses in the GI tract? Is there a link?
Yeah, that is an excellent question, and it's a really exciting area of research. Some of the hypotheses that we've been putting out are that bats may not be as dependent on services provided by the intestinal microbiome, and this may have influenced the evolution and adaptation of their immune systems. So perhaps they're not forming such tight links and relationships at that epithelial barrier in the gut. And if that's the case, they may be exhibiting more generalized responses to symbionts that come through their system. This could actually impact the relationship they have with viruses. So if they're having a more general response and sort of keeping the microbes at bay, they may be having similar responses to viruses and sort of allowing them to be transient members of the community that pass through. There are several other hypotheses about why viruses in bats are so pathogenic when they do transfer over to humans. So I just want to make a couple of notes here. Bats don't actually harbor more viruses than other groups of animals. They're just very diverse. Bats are very diverse, and so they have a proportional diversity of viruses as well. That said, the viruses that do move from bats into other animals tend to be more pathogenic. And one of the reasons we think that might be is due to the evolution of flight, which produces a lot of free radicals, and this has led to some unique adaptations in bats that allow them to repair DNA. They have like unique DNA repair mechanisms that are different from other animals. And this kind of creates an arms race in some ways between viruses and the host. And so viruses that are evolving to infect bats are actually able to persist in much stronger ways than they would in other organisms. So when they jump into a different host, they find it a lot easier to infect and proliferate. Coming back to the role of the microbiome, yeah, I think I kind of said this at the beginning that we don't really fully understand how this is working, but I think it has a lot to do with their immune systems. I'm not sure if that really answers the question.
No. It's a great answer. But to follow up on that, do we see any significant differences in fecal microbiomes for same species, but present in different regions and continents? So I guess.
Yeah. So this is seen in humans. It's seen in our domestic pets. You can very clearly see signatures of geography and distinguish populations based on where they're located. That said, the functional profiles of these microbes of the microbiomes are very similar. So you can think of it as, whether the species vary from continent to continent or, coast to coast, the species or the strains might be different, which is what allows us to distinguish where you're from. But the function of the microbes is likely to be very similar within species.
Mm-hmm. Yeah, so I just want to add something. There's a question about dysbiosis index in different countries and continents. Yeah, we looked at many different countries and continents, and we actually don't see a difference in the microbiome. And again, because the index itself, again, we need to understand we're comparing it to a disease population. So again, there's a much larger shift in disease than you would notice in some country dependent. So we have different countries and we don't really see a significant difference. And again, maybe also in dogs and cats, I think many of them are on commercial dog food diets, so they're probably more similar in composition, whereas in humans, obviously, there's a huge impact on dietary differences between Asian countries or European countries or Western countries and so on. I think that's the reason. Diets. Let's talk about probiotics again. Gail, is there a good resource listing specific beneficial bacteria and the corresponding effects of those bacteria?
Oh,
Hence,
that would be, yeah.
Hence,
No,
would there be a, sorry, just to finish, hence would we know if a probiotic is more anti-inflammatory or immune stimulating?
Yeah, I think the best place to get that information would be from the manufacturer. I haven't seen a overview presentation for probiotics that are out on the market for pets. There's a little bit more information for the human products, but I've never seen one that goes through their immune stimulating or anti-inflammatory properties. So unless somebody else on the panel has seen that type of thing, it'd be nice if it existed.
Yeah, I'm not aware of. Maybe a follow-up question. How do you measure transient versus permanent colonization
That's
in probiotics?
Yeah, basically what you do is you feed the probiotic. You have to have a test to be able to recover the probiotic from the feces, and you have the test that's specific to that strain. And basically what you do is feed the probiotic, get the fecal samples for various amounts of time after you stop feeding it, and see when it disappears from the feces. So you can tell whether or not it persists for a long time, or if it's gone after 3 or 4 days after stopping.
To follow up on that, is it important, is the adhesion property important for probiotics? So that means, is it essential for them to be effective?
That's an excellent question. It's something that's difficult to measure non-invasively in vitro, or in vivo rather. So I would say we probably don't know for the probiotics that we have, whether there is adhesion or not in the live animal. I think it probably depends on what the end benefit and the mechanism of action of that particular probiotic is, whether it's acting by, secreting bioactive substances that are having the benefit, in which case then adhesion might not be so important. If it's competing with pathogens, it probably depends on how it's doing that. For example, SF68 produces bacteriocins, and it might not actually have to have adhesion if you have a probiotic that's competing for binding sites in the GI tract with pathogens, then it would probably be more important.
A follow-up question for Holly. Also, individuality in species, would this also affect probiotics? So do probiotics have to be species specific, do you think? Or can we use one probiotic that works in one animal or human and use it in other species?
Yeah, that's a great question. I know that there are a lot of products that are marketing things like bifidobacterium for canines. And I think something that's important to keep in mind is that these groups, when we speak about them at the genus level or even higher levels of classification, they can be incredibly diverse. Even if it's a specific species, you might have different strains. And so I believe, and correct me if I'm wrong, that it won't be harmful to your animal, to the dog or the cat, to give them, a probiotic that maybe is something similar to what we're taking, but you're going to have limited benefit if it isn't species specific, and taking into account also just the individual variation of different breeds or the lineages of these animals. I think that you can fine tune it. I think it's important to fine tune it. And I don't think we're there yet because we don't have enough information or data on how a lot of these probiotics are actually benefiting the host.
Gail, your thoughts?
Yeah, I think it's something like Holly was saying, there's a lot of information we don't know yet. I know most probiotics I've looked at have efficacy across a variety of species. So for example, SF68 was isolated from a human baby, but most of the efficacy studies are in livestock and in pets and in adult humans, so they can have cross species benefits. Probably depends on what the benefit is that we're looking for as well.
Great, so that's for Professor Lappin. Is every E. Enterococcus faecium SF68 the same? I found this strain also in products for large animals. I guess that's what Gail mentioned. However, there was a different NCIMB number. Can I trust in a probiotic called E. faecium only?
Yeah, now again, it'll be a great one for Gail to add on here,
Okay.
Just because of the registration terminology, et cetera. But certainly, as Holly's already mentioned, genus and species. there are strain differences. One of the questions that's interesting about SF68 is, as it stayed the same, over the 40 years, since it was initially isolated, that if it's strain SF68, then in theory, it would be the same organism, but you couldn't trust another product that just says Enterococcus faecium. It may or may not be even remotely related to that organism, strain, or its effects.
Mm-hmm. Gail, you want to add to that?
Yeah, I would just say, basically the same as what Mike said. SF68 is a registered trademark. So if it specifically says SF68, then it would be produced the same way that the SF68 is produced for pet products that we use in Fortiflora. But I know that from what we've heard from the manufacturer of that probiotic, that you can affect its efficacy by how you ferment it. So if you had a different Enterococcus faecium, it wouldn't necessarily work the same. And there are pathogenic, as you all know, pathogenic strains of Enterococcus faecium too. So personally, if I was feeding an Enterococcus faecium probiotic, I'd want to make sure that it was one that really is a probiotic and not just one somebody isolated out of feces and then grew up. And then it's like,
yeah, great.
Yeah.
So, Mike.
I just wanted to go back to the previous question about will the same probiotic give the same effects in an alternate species? I think since we don't really know, it's really puts it upon the companies that market them for sale anyway, that they show efficacy, in the target animal species for those of us that buy them or teach vet students that these are the ones that work. It is actually quite important, I think, to have target animal efficacy data. And just one of the ones I think I mentioned briefly at the end of my talk was looking at Bifidobacterium longum, 999, coming from a dog launched product and then applying it to cats and seeing some of the same effects. So I guess I would be hopeful that many of those organisms would behave the same, and at least dogs versus cats. Now, whether or not dogs versus dolphins or dogs versus Gale, could certainly be different.
Yeah, that's a great point. So we are already there's two major areas that I still want to ask a few questions. So one of them is diagnostics, and then fiber. Those are major questions. And a question for Dr. Steiner, for Jörg. What are the approximate specificity, negative predictive value, and positive predictive value of cobalamin folate?
If we only knew the answer to that question, there is no, I mean, the best studies that were done on this were done in would say the mid 80s. Davenport did some of them, and there's some other people who looked at that. And the best I can tell you is that the sensitivities and specificities in those studies were all around 50, 60%. Nobody, as far as I'm aware of, has ever looked at positive or negative predictive value. And clearly, it's not stellar, it can't be stellar with sensitivities and specificity of 50 and 60%. It is until the dysbiosis index, it was the most practical and best we had. But, clearly that is no longer the case. But yeah, unfortunately, we don't know that. Especially the low cobalamin has many other conditions, obviously, that would also lead to a low cobalamin, any kind of severe and long-standing small intestinal disease that involves the distal small intestine. A high folate's a little bit different, probably a little bit more specific, simply based on the fact that other than supplementation, so some owners may supplement and you may not be aware of that, will obviously lead to an increased folate concentration, but really nothing else biologically will lead to an increase in folate. Now, but then the question is, how sensitive is it really? And what level do you use? If you just use the upper limit of normal, let's say is 15 and you use 16, that's certainly not going to be very specific. But if, we see dogs and cats that have serum folate concentrations of 30 and 40 and 60, and well, that becomes a little bit more suggestive. But yes, I mean, those are not very good diagnostic tests. Their sensitivity and specificity are both lacking. And I wish we would have more data on them, but we don't.
Mm-hmm. To follow up on that, also a question for Dr. Steiner, but then also for some other panel members. Oh, sorry. When is it appropriate to run a fecal PCR panel for a dog with chronic enteropathy, and how should a positive result for Clostridium difficile or Clostridium perfringens be interpreted? Jörg?
Well, I think those are really, that's a hard question to answer, because I think what we see at the referral practice, we see cases that have already been dealt with for quite a while. And so we probably run more of those panels than you would need to do in a primary care setting. But personally, even then, I don't run the panels very often. I run them when I have diarrhea that doesn't respond to the traditional workup that Linda has very nicely laid out, and I go by exactly the same thing that she laid out, with the minor difference in antibiotics as we discussed. But other than that, it's exactly the same thing. And so if I have bloody diarrhea where I can't find an underlying cause that's chronic, I certainly will run one of those. But the other question is a even more difficult one. What do I do with a positive difficile or Clostridium perfringens? The difficile one, I really, very little evidence that would suggest that it's truly a primary pathogen in dogs and cats. So I try to look for another cause in those patients. So I usually do not recommend treating them. And perfringens, it really depends on the species, on the strain. So just any measuring a toxin is very important, and the only one that's really has shown any sort of what Stefan has shown is the NET-F that has some agreement with what disease is actually clinically significant and with bloody diarrhea, but all the others are probably not really significant, and most normal animals have Clostridium perfringens. Most normal dogs, normal cats.
I might just add to that, because also was addressed to me the question. So we have a very strong correlation between the presence of Clostridium difficile and abnormal hieranones, so low hieranones, abnormal bile acid conversion, which also makes perfect sense, because it has been very nicely shown from human medicine, but also in vitro, that they are really very strongly interconnected. So alpha Clostridium difficile is truly, also my strong opinion, in most cases an overgrowth bacterium, because you have a dysbiosis to the underlying GI disease, you have no hieranones, no bile acids, and that's we see it very quite often. There's a few case reports in in veterinary medicine, mostly younger dogs, where they confirmed the toxin per ELISA, but those are very rare cases. So just PCR would indicate more dysbiosis. In selected cases, you may, if you rule out everything else, maybe consider running a toxin ELISA to confirm that it might be the rule, but it's very rare. Since we're running out of time, psyllium and fiber, there was a lot of question. Linda, how do you determine a starting psyllium dose? And there was just that the 5 milliliter twice daily was a confusion, I think, to the listener.
Well, as I said in my lecture, there are very few studies on fiber in diseased animals. There are quite a few studies in healthy animals, but very few in diseased animals. Still, we know that fibers can be very beneficial. So the dose I found somewhere, I think that was in the very old Stromback's manual of small, well, whatever it was called. It's not available any longer. It was sort of the Bible for gastroenterologists in the 90s, but it's not available any longer. But there they said 1 to 6 tablespoons of psyllium per day. So that's a huge difference. So I usually start quite low, and when I start, I tell the owners that most of these dogs will respond, but some will actually get worse. So that I usually start lower than that. So that border terrier I showed in my talk, she got 10 mils, which is 2/3 of a tablespoon per day to start with, just to see that they don't get much worse. And then you can taper up the dose. And usually if they get more than they can actually tolerate, the feces will be like a corkscrew appearance, which will fascinate the owner a lot. But if you decrease the dose, then they will get back to normal feces again. So you start somewhere, and then you can taper it up.
Mm-hmm. It's a follow-up question for Jörg. Would you try psyllium before tylosin?
Well, psyllium is sort of a very special fiber, and I think it's important to hold those two different, all these different fibers apart. The psyllium is a non-fermentable fiber, so that mostly has an effect with colonic disease, versus, if you have a fermentable fiber like fructooligosaccharides or some mixture of, whatever, inulin or whatever, that has a much bigger impact on the microbiota, as it's a substrate for microbiota nutritionally. So I don't use psyllium very often, mostly with colonic disease, isolated colonic disease, much more likely to use a prebiotic fiber, so a fermentable fiber.
Mike, I know you have some studies that you presented at ACIM with psyllium. Can you give us a background?
Yes, the most recent psyllium studies from our group have actually been with the Fortiflora SA product and the amount of psyllium that's added to that probiotic, and we'd shown potentially lessening severity of clavamox diarrhea, as well as using it for treatment of clavamox diarrhea. And then most recently with mycophenolate, when we get into treating those IBD and other immune diseases with immune suppressive agents, mycophenolate runs in that 20% diarrhea rate. And so I have not done psyllium alone, but probiotic plus psyllium, we were able to lessen severity of diarrhea in those mycophenolate dogs. So it becomes something to consider as we're treating our IBD dogs, is that could we perhaps have a positive effect on the microbiome and lessen toxicities of some of the drugs that we're using to treat that condition.
Since we heard today that a lot of things change over time, Stefan, what is the current thinking about fasting dogs with uncomplicated diarrhea for 12 to 24 hours?
These were all great questions, and this is also a good one. And yeah, actually, I think that this is also that we feed them earlier, and I know there are some studies also from York showing in parvovirosis cases that the barrier function is improved by early feeding. And I think nutrition for the enterocytes is also very important. So not giving diets to a dog with acute diarrhea is maybe only cosmetic, so that nothing is in the GI tract. But I would not give too much food at this time, of course, because we know this from ourselves that if we eat too much, that's maybe causing more problems. But I need small amounts frequently, and of a highly digestible diet is very important for the enterocytes, and maybe some fibers for the colonocytes as energy source. I think that's important.
Mm-hmm. Quick question to follow up, again, infectious agents. You mentioned, Stefan, you do not treat Campylobacter species. What about Campylobacter jejuni?
Actually, that's my philosophy. I do not treat a fecal panel. I always treat the patient concerning antibiotics, And for example, if I have a patient with a potential enteropathogen, I would maybe in this chronic disease and stable, I would rather consider to change the diet, maybe probiotics or fecal transplants to displace this pathogen or potential pathogen. But concerning antibiotics, I always treat the patient and I look for the clinical signs. So if I have an inflammatory response, if a dog is not responding to the diet and all other treatment options, then and it has an inflammatory response also on blood work, then in these cases I would treat with antibiotics. But I do not treat if I see, for example, Clostridium perfringens or Campylobacter or Salmonella, because in dogs there might be a bacterial strain without creating any problems to the dog.
As a follow-up question to Holly, how is Clostridium perfringens classified? It belongs to the Clostridium genera, but is not considered a beneficial bacterium.
Yeah, I think that this speaks to my earlier comment about the classification and taxonomy of these microbes. There's a lot of diversity below even that species level and the strain level. So Clostridium is a huge group, and many of the species that are in that genus have very different modes of action and different metabolic pathways that they utilize. And so you can't just say because it's a bacterium in this group that it's necessarily good or bad. I think we were thinking about these as whole communities too, and some may be pathogenic. It may also be context dependent. So, I mean, perfringens is actually frequently found as a part of the regular microbiome. It has more to do with the relative abundance of this microbe, probably in many cases. And you may find a positive PCR test and you may see that it's there, but it doesn't necessarily mean that it's causing a disease.
Mm-hmm. There's a question follow up on that, Holly. Maybe Mike knows more about also something. The relationship between intestinal worms and dysbiosis. What do we know about that?
That's an interesting area. I don't know much about it, but I think that there is a lot of research showing that the immune response to helminths or other intestinal parasites may have an indirect impact on the immune response to microbes as well. And I think there's even some evidence to show that helminths can directly influence the microbiota through their own metabolic pathways. Maybe someone else knows more than I do about this.
Yeah, I can't add much to that, but I would like to go back to the Campylobacter issue. We have had multiple drug-resistant Campylobacter going around the United States for over 5 years now that sickens owners as well as animals. And so it is quite important to remember that oral antibiotics and perhaps even parenteral can select for resistant strains of bugs that we just kind of routinely thought we could give them Altylosin or other drugs to, and then affect people in a One Health fashion.
Yeah. On that note, with the One Health, I think people here are probably aware that research has shown that our domestic pets and the owners often actually share microbes in the home environment. So we have to really think about how what we do with our pets or what we do with ourselves is moving in both directions.
Mm-hmm. Linda, you had some comments?
Yeah, first on Campylobacter, that if you infect 8-week-old puppies with Campylobacter, half of them will develop self-limiting disease. And it's very, very common especially for dogs under the age of 2 to have Campylobacter jejuni without having any clinical signs at all. So just because we find it doesn't mean that it's actually a problem. And secondly, there was a really nice paper that came out just recently on Giardia, where they had dogs that had very little symptoms or clinical signs of Giardia, but when they followed the microbiome over several months, they could show that they had a dysbiosis and it actually got worse over time, and they followed them for a few months. And so at least for Giardia, there seems to be some evidence that it can create dysbiosis.
Yeah, I saw this paper and I just want to highlight one thing that we, again, need to be a little bit careful, because many microbiome papers really just compare small effects. So that's important to always highlight. So when we see something cause a dysbiosis, we need to be really careful in overinterpretation. Doesn't mean that there's no changes, but again, many of those papers don't put this in a big perspective of what is the range of normal. So it can be a small shift. So I'm gonna just kind of highlight that. A few questions.
Jan, I think our time is coming to the end because it's, the discussion about microbiome can go on and go on because it's such exciting topic, and I really apologize to interrupt that. And but it's getting even more exciting because we have participants who are attending this conference from the different parts of the world, and we have such a great international faculty that is answering those questions and sharing experience. So I really hope that some of those questions still can be addressed tomorrow, because we have tomorrow another day. And not even maybe more exciting program for tomorrow, and those questions could be there. Also, you could visit the purinainstitute.com, where we designed specifically for veterinarian healthcare professionals the Purina Institute Microbiome Forum, a platform where you can find the foundational knowledge on the microbiome, where you can find the proven nutritional interventions, and that may help you in your clinical practice. And with this, Jan, if you have something to say, and then I come with certain announcements which are important for the day tomorrow.
No, I think thank everybody for really a lively discussion. We have a broad range of topics. We have a broad range of expertise, and I think it's really great. I think, as you can see, we know already something how to bring the microbiome into context. I guess we all probably agree that prevention is the way to go. The microbiome is an important organ for health benefits. So I think at this stage we are can we prevent it? That's probably the easier way to go. We still have to learn much about how to bring it back to normal. But I think I really love all those questions. Thank you, everybody, for bringing those questions. Sorry that we couldn't answer all of them. We're going to try to answer a few by email or by text. Some of those questions, especially about fecal transplants or microbiome dysbiosis, is going to be hopefully answered tomorrow, because we have a lecture about fecal transplantation tomorrow. And I'm hoping that you can join us back tomorrow.
Yeah, thank you very much again for everyone. And I know that it was a little bit too much time to ask from you. For those who was not able to see all presentations today or want to revisit it again, you can find them on the same platform, because they will be, all lectures will be available on demand. Also on the place which calls resources, you can find the list of the references that have been used during the presentations today. And another important point, that you can find the downloadable decks that has been presented today. If you go to the agenda and you see the names of their speakers and the titles of their presentations next to that, you will see the downloadable slides, which I believe would be very helpful. And with this, I really appreciate all your time, all your effort, all your knowledge, and the passion to talk about the microbiome, and looking forward to see you tomorrow. Thank you very much.